And now, please, the director of the Mendel Museum, Mrs. Křížová, for first speech at our meeting.
Thank you very much. Good morning, ladies and gentlemen. It's my very big honor to be here at your symposium. Welcome in the Mendel Museum of Masaryk University. Welcome in the Augustinian Abbey, the birthplace of genetics, and also the place where Gregor Johann Mendel lived and worked for more than 40 years. It is my great honor to present here the story of a humble genius, the story of Gregor Johann Mendel. And we can start. Gregor Johann Mendel is probably one of the most important names in the history of biology. I would say you'll probably agree with me. He's famous especially for his research and experiments with pea plants, but not many people know that he was really a Renaissance person. Actually, 2/3 of his research is devoted to meteorology. And he was a modest genius whose discovery was actually far ahead of his time. He's famous all over the world, but not in the Czech Republic. You can see I put a question mark here. And it was probably in the history, in the past, where there were communist era and communist regime. And Mendel wasn't very popular here because he was a Catholic priest and abbot. And he was the founder of genetics, the science which also wasn't very popular with communists. So, for example, when I was a little girl, or when my parents were young, they didn't learn about Mendel. You now wouldn't understand why, but I really believe and hope that it has changed because last year we celebrated a really special anniversary, 200 years of Mendel's birth. And in Brno there are many events. Our museum opened 2 new expositions, and there was this big international genetics conference. So now I believe that Mendel is known not only in the world, but also in the Czech Republic. As I'm not a geneticist, my talk will be focused on more on historical and cultural background. So I will discuss questions such as what were the key moments in Mendel's life, what relationships and conditions shaped him, and what were the basic qualities of Mendel's personality. So the story begins in Hinčice, in the northeast Moravia. I prepared a map for you, for those who are not from the Czech Republic. We are now here somewhere in Brno, in the South Moravia, and Mendel was born in Austrian Empire in 1822. So last year, as I mentioned, we celebrated 200 years of his birth. Mendel was born, according to the records in the church, on 20th July. However, Mendel himself celebrated his birthday on the 22nd. It hasn't been revealed the right date, so maybe in the future we will see what was the correct date of his birth. You can see here his native house in Hinčice, and also this original entry in the register of birth. You can see the date, 20th July, Johann Mendel. Mendel was born to a German-speaking family in Silesia, to Anton Mendel and Rosina Švidli. They had 5 children. However, 2 daughters died in early age. So Mendel had one older sister, Veronika, and younger sister, Rosina. They had a very nice relationship. For example, when Mendel was in financial problems and had no money for his studies, it was his sister, Rosina, who helped him, and she gave up part of her dowry so that Mendel could finish his studies. Later, Mendel paid it back when Rosina had 2 sons studying in Vienna, and he strongly supported their studies. So Mendel was christened Johann, but at the age of 21, when he entered the Augustinian Abbey, he took the name Gregor. Mendel was the only son, so he was to inherit the farm. However, since his early childhood, he was very talented boy, talented students. And here there comes a key moment in his life and key persons who influenced him. It was his teacher, Makita, and the parish priest, Schreiber. They recommended Mendel for further studies. So Mendel studied at Piari School in Lipník, and then at the grammar school in Opava, and then the Philosophical Institute in Olomouc. However, there were several misfortunes in a quick succession in Mendel's life. In 1838, it was his father who had injury in forest. That's why Mendel couldn't. That's why the family couldn't support Mendel in his studies anymore. So Mendel was 16 years old, and he had to make a living. So this caused many stress and stressful situations for him, because basically he had very little money for living. He started to teach his less talented classmates, but this money wasn't very strong for him. And he mentions this situation in his autobiography from 1850. And we can learn from this autobiography that he happened to be in a pitiful position of having to provide for himself, but that he managed to earn at least so much through private tuition that he could live an indigent life. So what happened next? The financial situation was very bad for Mendel. He also got ill from this stressful situation, and he had to spend 1 year in Hinčice with his parents. And in 1843, here we have another key moment in Mendel's life. He was recommended by his teacher, Friedrich Franz, to the Augustinians here in Brno as the most talented student of the year, and also for his unique character. And Mendel describes the situation and the circumstances also in his autobiography. And he mentions that it was the circumstances which decided about his choice. It was, of course, the financial situation, but also his big desire for knowledge and education, which he had really from his early childhood. And he gets to the Augustinian Abbey here in Brno, which was really the center of education, the center of knowledge in Moravia, I would say in Central Europe. Probably he couldn't get to the better place. And he was received by Abbot Knepp here in the picture, a key person in Mendel's life. I would say he was kind of a mentor for Mendel and supported his later research and his work. Here you can see the Mendel's refectory. Here, the same place as we are now. Now it has the Secession decorations, which was made after Mendel's death, and you can already see it here in this picture. This place actually was the refectory, the place where Mendel and other friars used to dine. So Mendel gets to the abbey. Here you can see the Augustinian library and the basilica. You will have chance to explore it on Thursday in our guided tours. And Mendel had really great place for studying. This Augustinian library has about 27,000 volumes, an amazing number of literature which Mendel could study and go through. And for example, he started to study such interesting languages such as Greek, Arabic, Syriac, Chaldean, or language of ancient Sumer. So you can see his theological study examination certificate. So he studied, of course, theology and other languages. So his desire for knowledge was really big, and he had great conditions here. And he was always interested in the natural sciences, so that's why he, in his first years in the order, besides the mandatory study of theology, he studied lectures on tree cultivation and vine growing as well. So what was the monastery community of Mendel's time? Here in the picture you can see the Abbot Knapp, theologist, orientalist, plant breeder, as well as member of Agricultural Society, who promoted new farming methods in the abbey and monastery estates. Matouš Klácel, philosopher interested in botany, mineralogy and astronomy. Or Tomáš Bratránek, philosopher, also expert in German literature. Later he became a rector in Poland, in Krakow. And Pavel Křížkovský, the composer, church choir master, and teacher of the foundation, which here in the monastery educated young boys who were singing here. And one of them was actually Luiz Janoušek, the music composer, also famous for his operas. And actually it was at the same time here where Mendel was living here and Janoušek was here as a young boy. Later, when Mendel died, it was Janoušek who conducted the requiem for Mendel at the Old Brno Monastery in 1884. Now, let me tell you something about Brno, about the cultural and historical background. Brno was really a bustling and lively city in the 19th century. It was called also the Moravian Manchester for its textile factories. There was this tradition of wool, textile and industry factories. The connection to Vienna was actually very early, from 1840 or 1839. So you can see the picture connecting trains from Brno to Vienna. In Brno there was also the Francis Museum, which was the oldest museum in Moravia, founded already in 1817. And this society around this museum was really educated. It sort of substituted the role of a scientific academy in Moravia. Then you can see Redota Theatre, the oldest theatre building in Central Europe. It was the place where all these pomological and vinicultural, horticultural section of the Agricultural Society which organized these competitions and exhibitions. And Mendel was the chief advisor and member of judging committee from 1863. He himself was an exhibitor, but also member of the judging committee. Another place for these competitions and exhibition was Lužánky Park. I really recommend this nice place and quiet place in Brno, which you can still visit. This was actually one of the first public parks in Central Europe. After Mendel finished his theological studies, he became a chaplain at the Old Brno parish. His duty was pastoral service in the church and in the parish. So it meant that Mendel visited sick and dying people, and it was very difficult for him. He actually got ill again. His illness manifested again. And it was probably that strong because Abbot Knapp described Mendel's state in a letter to the Brno bishop. Looking at the sick and suffering, he was afflicted with an overpowering fear, as a result of which he became dangerously ill. At that time, Mendel had to stay in bed for 4 months, so it was probably a big shock from this situation. So therefore, it was decided again by Abbot Knapp that Mendel would leave this position and Mendel would become a teacher. And he went to teach to Znojmo, and it was a very nice period in Mendel's life. I would say he was really popular teacher, popular with his students and popular with his colleagues, and he really enjoyed teaching very much. He taught their Greek, Latin, German, and mathematics. And in 1815, he was asked to take an exam to get a full teacher's qualification at the university in Vienna, because Mendel wasn't a proper teacher, he was just an assistant teacher. So he tried very hard, he prepared himself, but however, he failed the exams. And again, it was Abodnap who decided that Mendel would go to study to Vienna to broaden his studies. And that meant that Mendel studied in Vienna from 1851 to 1853, so 2 years in Vienna, which were really crucial for his research, for his future experiments. He studied with such experts, for example, with Christian Doppler or plant physiology by Franz Unger. And Mendel learned their new methods of mathematics, methods of combinatorics and statistics, which he later used in his experiments. So he completed his university studies, but again, in 1856, he failed the exams. You could see, or maybe we could say this was a big failure, or Mendel himself must have felt probably depressed. But from our point of view today, and for genetics, it was probably the lucky moment, we could say, because from this period, Mendel didn't become the full-time teacher. He was teaching only part-time at Realschule here in Brno. You can see the picture of the school and teachers teaching there. But he had more time for his experiments. He conducted for about 10 years here in the Abbey's premises, in the Abbey's garden, right in front of this building, and right on the place where new greenhouse now is standing. And he was one of the first biologists to apply mathematical methods to biological research and examine for more than 27,000 pea plants, which is really amazing for me. He made conclusions that are now known as Mendel's 3 laws of heredity. You all know, definitely, so maybe you can just skip this. And however, during his life, he wasn't presented as the founder of genetics. And when he published his famous work, Experiments on Plant Hybrids, in 1865, he met with very little response. It was only 16 years after his death when 3 scientists, you can see here, it was Karl Korrens, Hugo de Vries, and Erich von Czermak. They came to the same conclusions as Mendel and confirmed that it was Mendel who was the first, and titled him the father of genetics. And here is the garden you also went through in the morning. You can see how it used to look before, at the end of the 19th century, the beginning of the 20th century, and how it looks now. You can see special pea plants we grow here, really enormous plants, sometimes get really big. So we will see how big will be this year. And the greenhouse, which is now standing already, and you will have coffee breaks there, was built, the first one, the original one, was built in the 1850s. You can see the original plan of the greenhouse. And here, the picture shows the greenhouse from the 20th century, like in the first half of the century. It was severely damaged by the tornado in 1870. And it was demolished in the early 1960s. Some people can maybe see Mendel as an isolated monk among the peas in the garden and locked in the walls of the Abbey garden. But the truth is that Mendel actually probably liked traveling, because he traveled quite a lot, especially in 1860s. For example, he visited London, he visited Germany, and he went to the World Exhibition in 1862 to London. Here you can see the photograph from Paris with all the travelers who traveled by the so-called pleasure train from Vienna to London, to Paris, and then by boat to London. Next trip there was to Italy, where Mendel visited the Pope, and the third journey was to Germany, where there was the Bee conference. Mendel was really a polyhistor and a Renaissance man. So firstly, he was Catholic priest and abbot. He became an abbot in 1868, and many people know him, of course, as the founder of genetics, the biologist, but 2/3 of his work is devoted to meteorology. He was also the first person to describe a tornado, as the first scientist who described a tornado which was here in Brno in 1870. He was a keen teacher and a beekeeper, pomologist, and he even became a director of a mortgage bank. And also we could say he was an astronomer, as he watched the sunspots. Here you can see his microscope, which we have also in our exposition in the museum. And here also the famous picture you also saw in front of this Mendel's refectory. At the end of his life, he was said to say that although I have had to experience many bitter moments in my life, I must gratefully admit that the good and pleasant ones have prevailed. And the next statement is that my scientific work has brought me much satisfaction, and I'm convinced that I will soon be recognized by the world. And he really was. Last year, because there was this anniversary, important and special anniversary, Masaryk University scientists, under the direction of Professor Šárka Pospíšilová, conducted a laboratory research and analysis of samples they had exhumed from the Augustinian tomb in the Central Cemetery in Brno. So these samples were taken from items in Mendel's estate at the Mendel's Museum. You can see the scientists just working in our office, examining the items of Mendel, which Mendel used to use in his life. And the mitochondrial DNA of one of the hairs taken from a book of astronomy was then successfully matched with the DNA acquired from Mendel's presumed remains. And the team reconstructed about 99% of Mendel's genome. They also confirmed that Mendel had a genetic predisposition for heart arrhythmia. They also specified the height and length of his feet and increased brain capacity. As I mentioned at the beginning, last year we had this Mendel Genetics Conference here in Brno. You can see here the keynote speakers, the Nobel Prize laureates, for example, Sir Paul Nurse or Professor Thomas Čech, and here I'm with Ada Jonath, who visited also our museum. And you can see the sections of the conference here as well. A Mendel Museum opened 2 new expositions last year. The first one is devoted to genetics. You will have a chance to go through it on Thursday. It's a modern exposition with interactive parts. You can see Mendel himself just exploring DNA model. So we have also a program, the historical excursion to the Mendel's time. So we have a visitor, very important visitor, Gregor Mendel himself. And the second exposition we open is Mendel's room, the authentic place where Mendel lived and worked before he became an abbot in 1868. And Mendel Festival in 2022, it was a very big event for general public, for families, for children. We had this historical excursion. Here you can see Mendel celebrating his 200th birthday with pea cake. And finally, I would like to play a video just to sum up the facts about Mendel's life. I hope you will enjoy. This video was actually made also for Expo in Dubai, so that's why there are some subtitles in Arabic, but it's spoken in English. It was presented by Hvězdárna and Planetarium Brno and Czech TV. And I hope you will enjoy. So thank you for your attention. And now, please, let's play the video.
From the union of Anton Mendel and Rosa Mendel, 3 children were born. The eldest, Veronica, the youngest, Teresa, and on the 20th of July, 1822, I, Johann Mendel. Already during childhood, the first questions troubled me.
Father, how come plants present us with so many colors? Father, how come they have all kinds of shapes?
Well, I don't know, my son. And so I set out to schools in search of answers to my questions. But at the grammar school, some bad news reached me. Dear Johann, father seriously ill. You cannot study anymore. Your mother.
Johann, so you don't have to leave your studies. Here I am handing over to you part of my dowry.
Mendel, in order to carry on with your studies, my advice is enter a monastery. And so, at the age of 21, I joined the Augustinian monastery in Old Brno. And I became Gregor Mendel. At long last, I began to focus on my experiments. I chose an ordinary pea plant for this purpose. I always based my experimenting on extremely careful observation and mathematical analysis and statistics. And thus, I managed to predict what will happen when plants are hybridized. That hereditary elements, for you today, genes, are passed on from one generation to the next. The results of my research I presented under the title Experiments with Plant Hybrids at the Natural Sciences Society in Brno. What was he speaking about? I didn't understand any of it. At that time, everyone was fascinated with Darwin's new theory of evolution. No matter. 3 years later, I became the monastery's abbot, and no longer had much time left for pea plants. In the monastery's garden, I devoted myself to the crossbreeding of bees.
Ouch!
Also, to meteorological readings. And I was the first one ever to scientifically record a whirlwind, or, in other words, a tornado. And I kept looking higher and higher, and began to study sunspots. Oh well. More than enough work for one lifetime, eh? And so, as a result, I became ill and died on the 6th of January, 1884. But have no fear. 16 years after my death, my work was rediscovered by famous scientists, and they proved its validity.
Thank you for your attention. So if you have any questions, do we have still time for these questions?
Thank you very much for your speech. I am glad you are speaking not only curriculum vitae, but also the history of this place, history of Brno and south of Moravia. And now I would ask you if you have some question for the director Křížová. Nobody? Sometimes at the discussion in the other places. Thank you.
You're welcome. Have a nice day here at the Indonesia.
Thank you. It was first excellent speech, and now I would like to introduce this Mrs. Markéta Rine, which yes, please sit down here. And I would like to invite first speaker for our agriculture program. It's about history and evolution of the forage production at the Czech Republic. The author is the Mr. Nedělník, Dr. Nedělník, but he is illness and he is now in hospital and he cannot be here to speak. And we can change, and I would like invite Mrs. Koláčková, which take his speech. Thank you.
Thank you very much. So welcome everybody. Good morning. Unfortunately, Dr. Nedělník couldn't make it today, so you are stuck with me for about 15 minutes. It won't be a full 40, unfortunately, because that is out of my capacity right now, but at least it will be something. I will introduce you to forage farming, forage production of this century and the last. In the beginning, basically, Dr. Nedělník wanted to talk about the difficult situation that we faced as Europe and the whole world, basically. In last couple of years, we faced many challenges. It wasn't only about the security of this health and the security of populations, but also about food security. And that is a topic that is discussed widely in scientific circles. I think that the social isolation, as well as the Russian invasion on Ukraine and difficult political topics, are not really something that we should focus on today. We should do more positive topics. So maybe let's go into the last decade, maybe. Basically, at the same time that all of this was going on, those negative things, we got some positives out of it. The European Union started implementing the Common Agricultural Policy. It will be from this year, 2023, onward, that we will see some different changes in agriculture, hopefully for the better of the nature as well as our health as a population. Fodder or forage species have always played an important role in Czech agriculture, but also in Europe. And I believe it will be the same very important role in the future. This goes for both arable land as well as permanent grasslands or semi-permanent grasslands. In the Czech Republic, fodder has a very long and rich tradition in not only fodder production as a biomass production, but also seed production and plant breeding. I think that this high level of cultivation and also plant breeds and legume breeds is due to erudition of our farmers and plant breeders that has a rich history. You heard a perfect presentation about Mendel's life. The main three legume species, the main two rather, are the red clover, in here, red clover, alfalfa, and from the maybe lesser known but quite important for export purposes, it's a crimson clover, which serves a very important role as a pre-crop. The spectrum for legumes is quite a bit smaller in terms of species than it is for grass species, but we have many different varieties that we can use to adjust to different conditions. The term non-market crop still persists because the economic appreciation for the for these legumes only comes after the livestock production. Something with this figure. I'm sorry, this is not translated, but I will tell you at least. So the light green curve basically are the acreage of perennial forages on arable land. The red one, the red curve, is the numbers of cattle that are basically that we have in Czech Republic in the last, 40 years almost. And the dark green ones are permanent grasslands. And as you can see, the number of cattle goes rapidly down in the last 20 years. Before, in the 20th century, it was almost 3 million cattle head count, but nowadays it is only 1.4 million, which makes it about 0.4 cattle per hectare of land, registered land. And this goes hand in hand with the perennial crops on arable land because they are closely connected. We need to make a feed for animals, right? I know it is quite difficult to see for especially breeders of cattle to see this decline, but the dairy production had gone up significantly, so maybe there is something to take from all of this. Some interannual fluctuations were in terms of permanent grasslands, but there is no such significant decline or incline basically in acreage during the last 50 years. Depending on the actual average annual feed ration for cattle in Czech Republic, about quarter is from perennial fodder from arable land. A bit less is from forage for grasslands, mainly pastures and meadows and stuff. But most of this is from silage or conserved feed. In Czech Republic, mostly corn. I think that is the same for all over the world. This shows that the importance of perennial fodder is quite huge, and we have the legumes that have indisputable value, not only because of the production of quality feed that is rich in nitrogen and or crude protein, vitamins and minerals, but also because it has very positive effects on our soil. By having a symbiosis with nitrogen-fixing bacteria, we have a very positive melioration effect in the soil, as well as preparing the soil for after crops, basically. Legumes basically work as weeding crop. We don't have to use as much chemical herbicides. It depends on the location, of course. So, from those kind of values of legumes, it is also clear that we need to work on some stuff. We have very many different genotypes that we work on in terms of biotic and abiotic stress tolerance. Mostly, Czech breeding is focused on the abiotic drought tolerance, but also on biotic stressors, because there is the EU legislation that requires decrease of using of chemical and sometimes even biological pesticides. So we need to produce breeds or rather varieties that are resistant or at least resilient. The fact that in the last 10 years, basically, we were one of the largest basically, we created a lot of different varieties, resistant, non-resistant, for our market as well as foreign markets. We were one of the best, top 5 producers in alfalfa as well as red clovers. The export was much higher in 2015 in up to 2020. In the last 2 years, it went a bit down. This mainly was affected by alfalfa being more important into imported to Czech Republic than it was before. But still, red clover and crimson clovers are quite a huge export article. The production of red clover is about 220 kilograms per hectare in here, alfalfa about 150. For crimson clover, because it is such a huge export article, it is about 460 kilograms per hectare. These yields of seeds are mainly affected by harvest year, as I think all over the world, but also by the ability or actual farmers' knowledge of using of desiccants on the field to produce the seeds. Czech varieties are quite well known, I think, for the price, like very low price in comparison to usually American or Australian varieties, as well as the quality. We are quite a huge certified red clover producer, as I said. So what should the forage farming in the 21st century look like? Basically, it should be intensive, probably. It should be based on modern technologies, because we need modern technologies to establish the stands as well as harvest them for the full potential of new varieties. One of the main problems in Czech agriculture usually is stagnant or deteriorating soil quality. For this, basically, legumes are the best solution. If we want to enjoy the benefits and use the modern agriculture and the common agricultural policy, we will need to use the legumes as a stabilizing element in crop sequences. If we return those legumes, especially, but grass legume mixtures, more concretely into the crop sequencing, we will basically make the landscape forming element of our agriculture. The Czech Republic has the advantage of having such a great scientists and plant breeders that create a lot of different varieties, so why not choose from what we have available here? Clovers and other legumes, not only in monocultures, but especially in species-rich mixtures, can create, of course, good quality feed, but also an ecosystem that the nature can thrive in. Thank you very much for your time. It was quick, I hope. And thank you for being here.
Thank you very much for your speech, because the history and the question is if the 30 years is history, because you speak about only after Velvet Revolution. And I am the older man which remember collectivization. It is 60 years ago. At each village was the cooperative, and after that was this cooperative made bigger farmer, and now we have quite different size of the farmers, yes, and size of the cows, and very you can show the different numbers of cows, yes, but I have to say that the production total, our Czech Republic is the same. It's a very important point for our country, and the evolution with the production of the milk was very expressive, yes, and this is other side.
Definitely.
But I would like to ask if some from you have a question. Oh, please, you.
There is a microphone up here if you want to use it for your questions, so please raise your hand. But while you are thinking, I could ask, I noticed that quite big part of the dairy cow diets or the cattle diets is from corn silage. So do you see, is there going to be changes in that? Is the maize or corn silage still going to increase, or is there going to be more use of the grasses and the legumes?
That is a tough question. I wish I've known the answer. I think there are different farmers are testing the alternatives, for example, sorghum silage or different kind of like alfalfa mixtures. I don't think it's so common yet, maybe in like smaller farmers, but I wonder if somebody else has different opinion. But I think that for a couple of years it will be the same, but maybe slowly changing due to droughts. Like the maize has really low yields in the last few years because of the summer drought, so maybe that will be something to change it in the future.
Does anybody have some comments on this topic or some other
Please have
questions or comments?
I was not surprised that you told us the level of cattle in Czech Republic is very low, 0.4, I remember. So what is the strategy of Czech Republic's dairy farmers? Are there one part very intensive, and on the other hand in a semi-natural grassland? Or what will going on in Czech Republic in the 21st century in this case?
Wow, that is a tough question. I don't know. I think that the differences between very intensive and very extensive, like small local regional farms, will be basically more and more prominent in the following years. But I think there is a push to have some subsidies or to kind of have benefits for smaller farmers to make the regional sales better for the dairy as well as like meat. But I'm not really sure, I'm sorry. I don't have like a expert opinion on this. Maybe somebody else could add something.
I think we can also continue the discussions over the coffee. So next we will have coffee break in the glass house. But please be back here at 10:40 sharp, because we have some interesting lectures. We will continue with Luiz Ferrareto about the corn silage issues then. So now let's enjoy the coffee and keep discussing. Thank you. Welcome everybody. Let's continue our seminar, and the next speaker is Luiz Ferrareto. He's a ruminant nutrition extension specialist at the Wisconsin University, United States of America, and he's going to tell us about strategies to improve silage fiber digestibility. I guess it's mostly about maize or corn. Okay, the floor is yours. Go ahead.
Good morning. First, thank you for the introduction, as well as thank you for the opportunity of being here today. I was telling some colleagues this morning that I'm used with extension in the United States, so usually people take me to, a lot of different places for talks, But it's the first time I have the opportunity to present in a museum, in such a nice room like this. So thanks a lot for this, because, now I can brag with my colleagues that, I may be one of the few that had opportunities like that. But except for that, the topic that we were asked to discuss with you today is strategies to improve fiber digestibility in silage. So said that, we have some objectives for today's presentation. So we will try to review some indicators of corn silage fiber digestibility. And then we'll try to discuss some strategies that we can use to improve or change some of those indicators and consequently improve digestibility. Okay? We also will try to connect how forage quality affects cow performance as well as feeding behavior. Okay? It's important that all those three objectives here, they will be presented together. Okay? They will not be in this specific order. And then we also discuss a little bit, and re-emphasize the importance of kernel processing. So you may be thinking, Luis, why do you have kernel processing, right, in a presentation about fiber digestibility? You're going to learn about that at the end of the presentation, okay? So all the time we discuss fiber, right, we have to think first from the point of a dairy cow. And if we consider a dairy cow, I'm consuming about 25 kilograms of dry matter per day. A lot of times that cow will consume at least 7 kilograms of forage fiber, okay? However, as we start going through the sites of potential digestion for the cow, obviously the rumen is where we want that to be digested. From 7 kilograms, very likely that cow will digest about 2.5 kilograms, right? So that's actually less than 50%. Some cows will digest more, probably around 5. Some cows will digest less, sometimes only 1 kilograms out of 7, which is a very low number, right, for forage digestibility. But if we think about the total tract digestion, there is a little bit of compensation in the large intestine, but the numbers is still very low, right? So if we get good numbers, cows digest about 50%, right? So it's not efficient at all, right? And our goal is to be efficient. And if you go through literature, for every kilogram of digestible fiber, there is potential for 3 kilograms of milk production. So there's a lot there to consider, right? So when I think about fiber quality indicators, this is a very short list for that. It's of course a biased list as well, because as a scientist, I have my preferred assays, right? Those that I work the most and those that I see that may have greater potential to work. Okay, but you have to figure out those that work for you, right? So here I will discuss in more details this list in a little bit. But why do we care about indicators of fiber digestibility? So the practical implication of those numbers basically are intake limitation through rumen fill. It's a very common fact, right? Especially when we work with high-producing cows. The more milk a cow produces, more she has to eat, more likely to have rumen fill. Basically, indigestible material gets stuck in the rumen. She cannot eat anymore, she cannot produce more milk. It's simple like this, right? But it's also very important from the perspective of the establishment of high forage diets, right? We saw a presentation earlier today about different numbers of forages utilization. And in the United States, one of the things that farmers are doing more and more is emphasizing the potential of increasing the amount of forage in a diet. But the only way you can establish those diets is if you have very digestible forages. The less digestible it is, harder
Sorghum silage is less digestible than corn silage. Okay, that's just how it is. I really like sorghum silage.
Still have a long way to go to implement high forage diets with sorghum silage. Okay, that's just how it is. So we use those studies comparing those two crops to see. So if you feed a more digestible forage, what happens, right? So the comparisons that we have here is between cows-fed corn silage-based diets and cows-fed sorghum silage-based diets. So let's use this first study here as the example. So the cows consuming the sorghum silage-based diet consume 88% of what the cows consuming the corn silage-based diets did. And if you go across all those studies, those cows consume between 85 and 95% of what the cows consuming the more digestible diet. But you already knew that. I just showed you in the slide before the impact that that has, right? What we didn't know during that time, although I think Dr. Grant knows, what I did not know during that time was those cows consume 85 to 95%, and they spend 6 to 20% more time at the feed bunk. So let me repeat that to you. They consume much less, and they spend much more time at the feed bunk. What are they doing there? Right? I'll tell you what they are not doing in a minute. Topic, okay? And every 5 to 10 years, I receive a question, or researchers receive a question. So, are modern hybrids still susceptible to issues associated with maturity? So, some years ago, that's data out of University of Florida, okay, led by Dr. Bola de Zogan, and he planted a lot of different hybrids there during the summer, and he had 4 different target harvest maturities. And I will focus on very few things from this study. First, is starch increase up to 40%. We knew that, okay? This is true everywhere, okay? This is true in Wisconsin as well, okay? Our agronomists have very similar data sets. In vitro NDF digestibility decreases. But Luiz, the decrease is not as pronounced as for other factors, such as BMR, for example, right? True, it still happens, right? However, there is some trading yield, okay? So again, yield or quality, and you need to make a decision, okay? There's no correct answer. Every dairy has a different need, and the job of the consultant is to figure out what is the need and give the best advice, okay? The second potential change you can make in corn silage is related to chop height. So let's use those corn plants as an example, okay? And those values, they are arbitrary, okay? They may not be representative everywhere, okay? But let's think that this is the normal, okay? So usually, that's what you leave in the field. If you increase, you are leaving a little bit more. But pay attention to what you are leaving behind. You're leaving stock behind, right? None of the leaves are catch. The ears is still here. You're leaving stock behind, okay? I'm not gonna talk about this, okay? But this is something that happens in US sometimes, especially for research. They call topplage. You harvest immediately below the ear, and then there is a suggestion that you can go back to the field and harvest as a stocklage. My suggestion for you, don't do it, okay? Harvesting twice, one time, 2 times, costs twice, right? Costs 2 times whatever you have. It's not worth. There's no way you can pay for that. Don't do it, okay? But this may be worth for some farms, okay? So here's an average of 7 studies, okay? And the reason why we only have 7 studies here is we decide to put together all the studies after a review from the Penn State University, okay? And you can see the differences here. Those are centimeters, okay? Here it says inches, but it's centimeters. It's about 20 centimeters, like 50 centimeters. There is an increase in vitro NDF digestibility. There is a small increase in starch, less yield, okay? There is a trade, okay? You want better quality, you're gonna leave more material in the field, right? That's how it is, okay? It's not for everyone, but for some it could be an option, right? So we decide to put together a meta-analysis to try to predict that effect. So for every 10 centimeters that you increase, that's the expected response. And I want to point it out to one more thing that has to be considered. You are diluting more kernel, or the same amount of corn kernels, in less plants. So basically, you have more starch because of that, but you also have more dry matter, okay? So it's not because you're accumulating more dry matter, it's because you are leaving a wetter material in the field, okay? Why does that matter? It matters because dry matter is very important for fermentation. In this case, I'm using as an example, we had a study where the dry matter values were kind of low. So it didn't matter too much for pH or lactic acid production, okay? Even though we have a difference here, right? But just think like this. What if I harvest at 38 and by increasing I go beyond 40, okay? So you have to consider that as well. Is that what's gonna completely change fermentation? Not necessarily, but you have to consider, right? Another important topic that people discuss a lot in the United States is plant population, okay? By using plant population, there is a discussion about maybe you're gonna increase yield and you may improve fiber digestibility, okay? Potential effects on starch digestibility are not understood, and there is a lack of trials comparing plant population and cutting height, or shop height, together. So some years ago, we did that, okay? So together with Dr. Joe Lauer and Dr. Randy Shaver, we put together a study where we use 8 hybrids, 2 relative maturities, okay? So we had hybrids of short-term and long-term, or it's not long, it's actually mid. And these were the planting rates that we had, okay? You can see that there is a huge gap here, like it's very wide. And then we cut the 15 and 60 centimeters, okay? And we wanted to see, we know we will lose material if we increase cutting height. Will plant population compensate for that, right? So that was one of our goals, okay? And I'll tell you that it will, because I'm not gonna show any interaction for you today, okay? All I'm gonna show is the plant population effects. So except for protein, okay, which is negligent here, we didn't see any effect on nutritive value. All we saw was a change in yield of dry matter. And that's the amount that we had there, okay? So by planting more plants or greater seeding rates, you increase yield, okay? So what we saw was, perhaps, if your goal is to increase shop height, you can also consider greater plant population or greater seeding rates, right? Something we have been discussing a lot in the United States for hybrids and for plant population. We reach a time that the farmers, in addition to be all the different, professions that they have inside a dairy farm, it's time for them to also be researchers, okay? And I know it sounds terrible when I say like this, but I honestly think that it's time for dairy farmers to test those things in a small scale under their conditions. This is University of Wisconsin, right? A lot of our management may not be what you do. So what we have been suggesting is, you want to test for plant population, try a couple stripes in a field and see how that compares with the common plant population that you use for the common hybrid that you plant, right? It may not be the same response, but if it is, you can be 100% sure you can implement that, right? For shop height, it's harder to try that, of course, right? For hybrid, I suggest the same way. Plant 1 or 2 fields of this novel hybrid, right? Preferably, try a very good field and a terrible field, because then you see the best potential scenario and the worst potential scenario, because you need to know both, right? This year, we are living the dream, everything is great. But what if we are not, right? So that's what I suggest for you, okay? So changing topics a little bit, all the time we discuss fiber digestibility and high forage diets, people ask, but Luis, we have so many industry byproducts in the United States that are so digestible. Why if we use some of those industry byproducts instead of some of those forages that are less digestible? Is that all right? So let's try to compare. So we run a study during the summer. Okay, we had multiple goals. I wanted to understand that during the Wisconsin summer. For the record, the Wisconsin summer is very soft. Okay, we barely have heat stress. Maybe a couple weeks. Okay, but it's nothing compared with other areas. Okay, but we wanted to understand maybe if that affects heat stress on cows too. Okay. So we have 2 diets, a high forage diet and a low forage diet, and I will show you the diets in a second, okay? And we treated those diets with an additive containing Lactobacillus plantarum, Buchneri, and Saccharomyces cerevisiae, and basically those were inoculated directly into those diets, okay? So those are the diets that we had, okay? So all we did is we removed corn silage of the diet, and then we added soy hulls. And in order to make sure we didn't have the effect of the different nutrients, we kind of changed a little bit the high moisture corn and the dry corn, so the diets had similar starch and similar NDF, right? So what we are testing here is NDF from a forage source versus NDF from a byproduct source. That's all we are testing in this diet. And you can see that those diets are very similar in nutrient composition, okay? And if you are wondering, yes, crude protein is very high. Luis committed the mistake of using the new NRC to formulate the diet, okay? Don't do it, okay? So starch is very close, NDF is very close, okay? There is some variation there, of course, right? Those are 4 different diets. This is the Penn State particle separator. You can see that there's not a huge change, but Luis, you replace forages with byproducts. You don't have changes in particle size. Well, our corn silage is actually very fine, okay? So that's why we don't see the drastic change. But the question is, what did the cows tell us? So as expected, you have an increase in intake when you feed the low forage diet, right? That's an obvious assumption. Soy hulls are always more digestible than corn silage. And if you have a corn silage that's more digestible than soy hulls, it's because you bought a very bad soy hull, okay? It's not because your corn silage is great, okay? It's just how it is, okay? You can see that there is a small increase in milk production. However, there is no effect in fat-corrected milk, no effect in energy-corrected milk. And then we have fat concentration. We have a decrease with soy hulls. Our milk fat was not already great because we are in the middle of the summer, okay? Usually our cows produce 3.84% fat. During the summer we have this drop. Look what it did to that, right? So basically what we saw is, and somehow I didn't add here, yeah, I didn't add here for whatever reason, but the energy-corrected milk efficiency, which is energy-corrected milk divided by dry matter intake, was greater for the high forage diet, okay? So you cannot replace forage. Cows need forages even though they are less digestible than byproducts, okay? But increasing the quality of the forage, that goes a long way, okay? Of course that if you need to feed more byproducts for any given reason, you have to do what you have to do. But that's what we saw, okay? And then the other thing I want to show you is some of the respiration rates, okay? We didn't see much from a forage perspective, but the additive we use actually reduced respiration rate in the morning, which to us was quite interesting, right? Because there is potential to mitigate heat stress, okay? Of course, this is a one study. We are still learning why that happened, right? But I think it's very promising, right? The other factor that we saw related to the additive that I don't have data here, we saw a couple weeks during this study some benefits in TMR stability, right? So perhaps adding some of those microbes at feeding may have the benefit as well. Again, we are still learning about that, but could be some potential, right? But going back to the forage discussion, I know that those numbers look very, very similar based on 1 decimal place, but there is also an effect here, okay? And basically, for every single parameter, there is an increase when you feed byproducts in animal temperature, okay? And the reason for that is probably you're generating more heat, okay? Luis, is this small increase biologically meaningful? I don't know, but we have to consider because we tested that in a freestall barn, very well equipped with fans, okay? During a summer that is actually not that warm, okay? The question is, if it's very warm, I guarantee you the response will be much higher than that, okay? But again, I'm only speculating, right? I didn't test that, so be careful with that, okay? So the last part of my presentation, you know, you probably are wondering, so now is the time we're gonna learn how kernel processing affects fiber digestibility. And that's the time in the presentation that I will disappoint you, because I don't know how, okay? All I'm gonna tell you is starch is my favorite topic, and I know that kernel processing is the most important starch-related topic for corn silage. Probably here. This is also true in the United States. I'm Brazilian. I know this is true in Brazil, and I'm 100% sure it's true in other areas in the world, okay? So I'm gonna explain to you why do I think that kernel processing is so important. So many years ago, I was in grad school, okay? And we had a group of students working with us, and they asked Dr. Shaver during the time, can we run a study to understand better particles of corn kernels? And Dr. Shaver said, sure, whatever you guys like, right? Dr. Shaver had a nice collection of hybrids, you know, corn kernels from different hybrids collected at silage maturity. And the students decided to get 100 of those hybrids, and they separated in different pools and get 3 kernels for each of those pools. So 300 kernels they cut in half, 300 kernels they cut in 4, okay, and they use a small scalpel to do that, so, you know, very good exercise. So if you're considering going to grad school, you know, think twice, okay? in 8, 16, and 32 pieces, okay? And that's what we agreed. But they decided that they want to cut into 64 pieces, okay? So they asked Dr. Shaver, he said, sure, if you guys want it, but you don't have to, right? They decided they wanted to do that. I don't know why, okay? It took a lot of time. I'm very happy I didn't join them. I didn't cut a single kernel, okay? I'm just the lucky guy that presents that, okay? And that's what they did. But then they collect those kernels and they put inside those bags. If you never saw those bags, these are in situ bags, okay? Then we put the in situ bags in the laundry bags, and then of course I joined them for the most important part of the trial, okay? I put the bags inside the cow. This is me, okay? Believe it or not, okay? So we introduced that in the rumen of a dairy cow, and we leave there for 3, 6, 12, or 24 hours. And that's what we saw, okay? In addition to those, we had whole kernels. Do you know what happens to whole kernels in the rumen of a dairy cow? You're right, nothing. That's what it is. In 2 pieces, it's a little bit better. 4 is a little bit better, and so on, okay? Keep in mind that these are unfermented kernels, so it's a little bit different than what corn silage is, but that's what it does. And then based on that, we decide to test a couple different things, okay? First, I want to show you that kernel processing score and fecal starch are very well related, okay? So it just changed cow performance. But more important than that, did you know that starch digestibility increases over time that you let that ferment in the silo? Is it true for the intact kernel? So we run a study to do that, okay? So we collect the whole plants. The students separated into forage and kernels. And then the kernels, they either kept intact or they smash one by one just to open, right? Let's think about the rumen. Bacteria has to enter the kernel to attach and degrade. Pericarp does not allow that. Pericarp is the corn hull, right? So why this would be true in the silo? So that's what we did, recombine, ring silo, okay? A much easier trial than the previous one. It's still a lot of work, okay? And what we learned is the intact kernels didn't have greater digestibility with fermentation. The broken did, okay? Now, let's put aside the fact I'm a scientist, okay? Let me tell you what Luiz thinks about that, okay? I don't think the data is right, okay? It fits exactly what I thought, but it's not right, okay? I think that the intact kernel will improve a little bit in digestibility because there are enzymes inside the kernel that can break down those prolamine proteins that inhibit starch digestion, okay? So if I had to guess, I would guess that increases a little bit, but not to the same extent, okay? So if you need another reason to improve your kernel processing scores, in addition to the cow digestibility, is because if you want to get the full benefit of fermentation, starch has to be available to the bacteria in the silo as well, okay? So in conclusion, there are many factors that alter fiber digestibility in corn silage, and if you can reach the greater fiber digestibility, very likely there is greater intake, and that allows for the establishment of high forage diets. I didn't discuss that today, but forage particle size as well as digestibility, they not only drive performance, but they also modulate feeding behavior patterns. Remember I mentioned the cows are there eating for longer? This is also true if you have very long particles, okay? And last, it's a very simple message, break more kernels. With that, I would like to thanks again for the invitation to be here. Thank you for your attention. I'll be glad to answer any questions if we still have time. Thank you very much.
Thank you very much, Luiz, for your informative presentation, but I'm sure there is still something you want to know about corn silage fiber digestibility, so please use the opportunity.
Hi Lewis, Gordon Marley from Alltech. Thank you very much for a lovely presentation. Very, very clear. In Northern Europe, when we're getting into the Baltic states, sometimes we are harvesting corn silage at 28% dry matter, 29, 30% dry matter. What's your thought process on the processing requirements at suboptimal dry matters?
Yeah, that's a great question. I think that when we are harvesting, I think to achieve greater processing, we have to consider multiple factors, and obviously maturity of harvest is one of those, right? Usually 30% dry matter corn silage, the kernels are very immature and they are easier to break, okay? They still need to be broken, right? So you need to make sure you still use the cracker. Maybe you don't need to go as fine in particle size and as tight in kernel processing as you would need for a 35, 38% corn silage, right? However, there are two considerations that you have to make, okay? The first one is seed genetics is changing a lot the corn plant for good, right? If you go to the United States, you see that we have much healthier plants today, right? And this is outstanding for farmers, right? But at the same time we do that, we are actually creating a disparity in the genetics, right? In the dry down ratio of the plant and the dry down ratio of the kernels. So there are scenarios where we see very wet plants with actually harder kernels than used to be 10 years ago, okay? So from my perspective, you have to pay attention not only to the dry matter, but to the kernel milk line to make the decision you ask about. Because if you have a very mature plant, but the kernels are already very well formed, you have to make sure you are aggressive with processing, right? If it's very mature, then we can go back to the previous scenarios I described and be a little bit more flexible. One other thing that I think people have to consider about that, especially if they analyze corn silage processing score in a laboratory, okay? Very mature corn silage, the kernel do not shatter. They are smashed open, okay? And because of that, there are some seed companies in the United States, they use that to say that you have to delay harvest, right? That's not real, okay? That's a fabrication of the assay, okay? So what you have to do with that is, that smashed corn kernel that's very mature, it's extremely more digestible than that shattered, very mature kernel, okay? So keep that in mind, look into those numbers, because sometimes you may think that you have a bad processing with the immature values, and in reality it's just because those are smashed open, okay? I know this was not your question, but I thought I would mention because this always comes up for us in US, so.
Thank you for your presentation. It was excellent. My name is Silvia Orosz from Hungary. I have a question about the future of the maize silage in the United States. In dry continental region here in Central Europe, we had many problems with the climate change, the drought and the heat stress. Not the drought is the biggest problem, but the heat stress. The 40 Celsius degree for 1 week, it will kill the corn. So it is the first question. And the second question is, what do you think about replacement of maize silage or corn silage? Because we have new sorghum types now in Europe, BMR, PPS, maize-sterile sorghums with very good fiber digestibility. And we started to combine these sorghums with high moisture corn. So it is the second question. What is your opinion about it? Thank you.
Thank you for the questions. With regard to climate change, right, I believe we all know that's happening everywhere, right? I joke with my colleagues that Wisconsin is getting colder, okay, so I don't know if it's global warming the right name, but it's definitely changing and not for better, right? With regard to corn silage production, Wisconsin still does not have issues associated with that. The southern states of the United States do, right? Especially because they have a lot of issues with water, right? Availability and regulation. So there are many areas, especially Texas, New Mexico, that are actually already investing a lot in sorghum silage, not because they want, but because it's a need, right? So from that perspective, I expect sorghum silage quality to continue to improve, right? And we're gonna have to replace corn silage as needed based on that. This is also true for other forages depending on other issues. For example, for us in Wisconsin, there are areas that there is a lot of alfalfa winter kill. So the issue is actually how to have alfalfa, right? So this is definitely going on, and we have to adjust and adapt based on what we can, right? With regard to your second question, I agree with you that there are a lot of different options for sorghum, and there are a lot of BMR sorghum as well that is very digestible in the United States. And if you go back to the literature, even those studies that I showed from Rick Grant, if they compare with the BMR sorghum, intake was actually similar, right? So there is a lot of potential of using those. The issues associated with BMR sorghum are exactly the same issues associated with BMR corn. Produce less per area, is slightly less resistant to disease, right? So all the time you implement those technologies, you have to be aware of the pros and cons and do your best based on that. But you're absolutely right. There are different sorghum technologies available that can be utilized. And if I could suggest, and you really want to replace corn, that's the way to go, because with conventional sorghum, it will be a struggle. However, we are discussing the fiber portion of sorghum. There is one thing that is irreplaceable, which is the starch, okay? Sorghum starch is less digestible than corn starch. Sorghum berries are much smaller than kernels in corn, right? It's virtually impossible to break it, okay? We actually work in a berry processing score project for sorghum, right? Similar to the corn silage processing score. And it's actually very hard to even define which sieve to use, right? The initial development of that use a very small sieve, and when I look to that sieve, if you can break sorghum berries that small to pass through that sieve, you are a hero, because it's virtually impossible to do that in the field. So we actually increased the sieve, and even like this, we couldn't get good results. So if you have to implement sorghum, that's the challenge you have to think about. How do I break those berries? And it's not gonna be easy, okay? It's a necessary evil, right? But you're gonna have to understand that issue, so.
Thank you. I think we need to move on now, and thank you. Oh, okay.
Hello, Luis. We work with the digestibility a lot of time, yes. We started tested in front of Velvet Revolution. And after the revolution, we change, we more testing selection of hybrids, yes. It's very important to selection. And we cooperated from the 2009 year with Joe Lauer. Yeah. And I am very glad that you show his slides, yes, because he visited our meeting last 2 times and last meeting also. Your view is only for nutrition, yes, it's perfect, and I am glad that you are here. But we are the specialists for fermentation, and you advise dry matter over 40.
No, never.
No?
So, in my opinion, you should always focus on the more immature silage. My recommendation today is 32 to 35% dry matter, as far as your kernel milk line is between 2/3 or 3/4 of the kernel.
Yes, but the results was very good for the also for the milk production per hectare. Yes, for the higher dry matter. Yes, and that may be. I think problem with the hygienic quality with that, yes. Yeah. And I would ask you about the hygienic quality with the high dry matter, because when I am going to the farm, some foraging nutritionists which would like the higher content of starch, they advise higher diameter because there is higher starch, yes. Yeah. And I would like if you speak about it.
As a nutritionist, I can say that I disagree with those nutritionists. Yeah, it's very important to have more starch in corn silage because you can buy less corn, but there are some issues, right? First, corn silage has lower fiber digestibility if it's more mature. There is potential to ferment to a lesser extent, right? Particle size will be coarser, so packing and density very likely will be worse. You allow silage to stay longer in the field, so very likely you have more yeasts and molds that may be problematic later on, okay? Now let's go to the starch portion, okay? The corn kernels are much harder, so you don't know if you're gonna break it or not, okay? You have to be more aggressive, okay? Of course. Oh, you don't want my answer? Sure. So, corn kernels will be much harder, right? So much harder to break, right? The starch chemistry also shows that there is much more prolamine, so the starch is naturally less digestible. More starch does not mean more digestible starch. Oh, I saw a calculation one time that shows that the changes in vitro starch digestibility multiplied by starch concentration multiplied by yield show the same value in the field. Sure. Ask the cow what she thinks. I don't care how many kernels there is in an acre or hectare. Tell the cows that, right? And she's gonna tell you in the manure what she thinks. You're gonna have bird food waiting for you, and that's how it works, okay? I know that because that's a very common discussion in the United States as well. And obviously, the most important factor is not even starch. The real factor is, can I harvest enough dry matter yield? And that's very difficult, I understand that, I agree with you. So you have to find the sweet spot. But from a digestibility perspective, I guarantee you, the more you wait to harvest, more you have to lose. And I think every single trial available shows that. In Brazil, in the United States, I guarantee you if you start running those here, you see that as well. Maybe you already have, right? So I would be very careful with that. If you have to because of yield, I understand. If you want to because of starch, I would think twice about that. So.
Okay, thanks very much for this insightful discussion as well. But now we thank Luiz and we move forward with the program. We invite.
Focus on the nutrition, not much regarding the fermentation in silo, but more how the animal handle this. So initially, when you look at the literature, the typical literature, basically one summarize the fermentation end products. The only alcohol summarized is typically ethanol. I think there is two main reasons for that. First, because it's really important in quantitative terms, and also because there is not too much literature regarding other types of alcohols. So here in this table from Limingkang, different crops with typical range of alcohols. As we may see, alcohol are more present in silage that is more easy to ferment, like maize silage and high moisture, compared with grass and legume. But if you see at this value, it's considered very, very low with a situation we have in the tropics. Sugarcane silage, it's a very high ethanol silage, sometimes you can have more than 20% of ethanol in sugarcane silage. But also, I think here you can have sugar beet, also high in ethanol, and sweet sorghum, it's also high ethanol. Okay, but as I mentioned before, silage has not only ethanol. Here, a table from Dr. Weiss with 12 alcohols, different concentrations. Recently, the group from Antonio reported 16 alcohols in different silages in Italy. So again, ethanol by far is more important quantitatively, but silage also has other type of alcohols, and we're still learning how the animal thrive on this kind of alcohols. So where these compounds come from, we also are learning about that. Different microorganisms can produce ethanol, not only yeasts, as most time we discuss that ethanol comes from yeast, but there are different sources of ethanol in silage. But the parallel situation is the same microorganisms can produce different type of alcohols. So, and another concept here, you can see most of these microorganisms are considered undesirable on silage. So most time alcohols are associated with undesirable microorganisms. Said that how the nutritional models, they handle this information when you are formulating a diet. When you look for at least in my experience in Brazil, these are the main nutritional models used. The American NRC and the new version, the Cornell model and the NOR4. In all cases, there is no specific use of this information in the diet formulation. For instance, in the NRC 2001, the alcohols or all the fermentation products, they are included in the non-fiber carbohydrate fraction, calculated by difference, as it is in the NASN 2021, in the residual organic matter, calculated by difference. But it's only true if you correct the silage dry matter for the volatile compounds. If you don't do that, it will not include that. Okay. NOR4 recommends to correct the volatile compounds, and also use this information to estimate the relative silage index dry matter intake. At the Cornell, there are different fractions within carbohydrates, and alcohols especially are included in CB2 that is considered similar to pectin. So anyway, there is no specific information at all regarding alcohols or fermentation products in silage, although cows eat a lot of silage. Okay, moving to the next point of the presentation, perhaps a lot of you are thinking, you are talking about volatile compounds. Does the cow really eat these products? Or they are lost by volatilization? So here I will present you an average of 3 trials we did during my PhD, measure the loss of different fermentation products from the TMR. Even during the Brazilian summer, very hot summer, only 10 to 30% of the volatile compounds were lost from the TMR. 79% of the products were ingested by the cow. It means these compounds may affect cow performance. And how they can do that? Basically, by three different ways. By affecting feed intake, by affecting rumen fermentation, and directly by nutrient supply. Okay? So we will see the next slides where those different alcohols were studied. I will focus more directly on the cow performance, but these three different pathways will be responsible to change to explain the performance. So ethanol, in fact, is the most important. Here, a very known work from Dr. Wendby, comparing a control diet where the cows were another diet receiving pure ethanol. As you may see, ethanol increased intake and increased fat-corrected milk, increased fat, increased protein. Okay, but look, no effect on feed efficiency. No effect of feed efficiency. I will show you in a few minutes the importance of this number. Here a trial from our group, again control versus ethanol. Again, ethanol increased intake, increased milk more than 2 kilos per day, no effect on fat, no effect on protein. Again, no effect on feed efficiency. And why I'm insisting on the feed efficiency? When you look at the heat of combustion of alcohols compared with a typical dairy TMR, all alcohols, they have a greater heat of combustion compared with a dairy TMR. So when you supplement, as in the past trials, ethanol, it's really expected to have a greater feed efficiency than the control diet, but it was not the case. And why it happens? I will show you next slides. Here, 2 trials from the Danish group with transition cows, fresh cows. So they measure the metabolism of the cow, the recovery of alcohol in the portal vein, and as the lactation progresses, the dry matter intake increased. The content of ethanol in the diet was the same, so the intake of ethanol increased. But look, the recovery of alcohol even decreased. So even eating more ethanol, the cow metabolized more ethanol in the rumen. So less ethanol reached the liver of the cow. The same in the other trial. If you look at the portal flux, 113, the net hepatic flux minus 131, it means actually the cow metabolized much more than eaten. So there is no alcohol, virtually no alcohol in the arterial blood. So ethanol was not available at the tissues because it was metabolized before. And here, an in vitro trial where ethanol was supplied to the rumen fluid, and you may see a very clear effect of the ethanol being converted to VFA, mainly acetate, and beyond the acetate, ethanol was converted to methane. Okay? So this is why the cows do not really absorb ethanol. they eat ethanol, but absorb mainly acetate. And this is why the feed efficiency was the same in the trials. And this is led us to rethink this concept regarding the energy recovery, the gross energy recovery of 99% for the ethanolic fermentation. Because if the cows do not use ethanol to yield milk, it's not true. So if you consider that 25% of the alcohol is in fact absorbed, but 75% is converted to acetate, and then you should consider that energy, net energy recovery is much lower than the gross energy recovery proposed. So it means, although no negative effect on animal performance, alcoholic fermentation induces high dry matter and high net energy loss during ensiling, so it's completely undesirable. Here, a data set we combined to measure how ethanol explains gas loss in sugarcane silage. It's a very simple silage in terms of composition and also in terms of fermentation. And as you may see, there is a very, very tight relationship between ethanol and gas loss in sugarcane. And impressively, you can see the slope here is almost 1. It means 1 ethanol, 1 gas loss there. So the dry matter recovery is exactly 50%, and the theoretical proposed by in the table from Peter McDonald is 51. So exactly the same. Okay? So again, ethanolic fermentation is highly associated to fermentative loss, and then it should be controlled. So how to control this? Basically, at least in Brazil, we are using 2 types of strategies. One strategy, here again, an example from my reality for sugarcane silage, heterolytic bacteria. You can see here a trial with 4 treatments, a control, and these 3 treatments are different heterolytic treatments, these colored lines. And in the X and Y-X, you can see the gas production, the kinetics of gas production over time. And at the end of the trial, the control treatment resulted in 176 grams per kilo dry matter loss, and the inoculated treatments 49.9 grams per kilo dry matter. And why it happens? Because the gas production is stopped very earlier in the treated silage compared with the control silage. So one strategy to control alcohol is using these heterolytic inoculants. Another strategy is using antifungal chemical additives like benzoate, sorbate, propionate, or mixtures, and then here comparing a control, albuquinol and benzoate. As you may see, ethanol formation was 49% lower for the inoculant, but 97% lower for the benzoate. The dry matter loss was also reduced, and the soluble carbohydrates were more preserved with the additives. But look, the chemical additive was approximately the double of the efficiency compared with the inoculant. Okay, so there is more than one solution: a cheaper, expensive solution, a more efficient, a less efficient solution. Depends on the availability. You can you can take your decision. Moving to other alcohol, I just have one slide about 1-propanol. There are other literature, but I have no time to show. I will show you one trial from our team. 1-propanol was supplemented at 1% dry matter of the diet. No effect on cow performance. A trend of higher plasma glucose and lower plasma NIFA, as expected, because the propanol has a glycogenic effect, okay, but no effect on the animal performance. Another alcohol, 1,2-propandiol. There are here 2 doses in this trial. Okay, although it's right, the grams per day, the material was mixed in the TMR. It was not fed pulse dose. It was a mix in the TMR. There was a positive effect on intake, mainly on this first dose. No effect on milk yield, but a large effect on body weight gain. This is probably occurs because the 1,2-propandiol may induce insulin resistance, okay? And this is why it has been used to treat ketotic cows after the parturition. And also it has been thought that silage rich in 1,2-propandiol could be used in such a way. But there are 2 concerns about that. The first is silage 1,2-propandiol is not fed in pulse dose. So it's completely different when you treat a cow with a pulse dose of 1,2-propandiol. There is a trial from medicine showing that the only benefit was when the cows were treated as pulse dose, not mixed in the diet. Another concern is 1,2-propandiol concentration is positively correlated with acetic acid in silage. It's not perfect. There is a large distribution, but there is some positive correlation. So silage rich in 1,2-propandiol is probably also rich in acetic acid, okay? And depends on the concentration of acetic acid, you may see some intake depression. As you may see in this trial, acetic acid in corn silage 2.85 in the control, 4.41 for the treated, and a trend of lower intake and lower milk yield. Okay? But this effect occurred basically because the acetic acid in high concentration depressed the intake during the first meal. Right after the morning feeding, the cow ate less. And this explained the lower intake. There is a high correlation between the intake in the first meal with the dry matter intake along the day. However, this depression effect on intake is not always observed. For instance, a trial from Limingkang with alfalfa silage, even with a higher concentration of acetic acid in the treated silage, no effect on intake and a trend of higher milk yield. So now we are in a situation where in some trials high acetic acid depress intake, why in others do not. And then we performed a meta-analysis to try to understand that. And then we organized all trials available where there was no malfermentation. This is very important because almost all literature regarding acetic acid, there is a co-linearity between pure fermentation and intake. So it's impossible to attribute that to acetic acid based on the past literature. So then we decided to choose only trials where the silage was well fermented. How? There are two ways. One, where acetic acid was induced without a pure fermentation. How? Using a heterofermentative inoculant. And the other is if acetic acid was supplied in the diet, pure acetic acid. Okay? So the gray diamonds are inoculated with heterolactic, and the white are supplied with pure acetic acid. And as you may see, there was a broken line response. So during the first phase, there was a slight depression, but actually not, the slope was not different from zero. It means from 0 to this point, the cow is still eating the same. After that, there was a steeply decline on intake. So, as you may see here, when the acetic acid in the diet, not in the silage, the amount of acetic acid, the concentration of acetic acid in the diet will depend on both the concentration in the silage, but also the inclusion of silage in the diet. This is why, for instance, let me show you. In this trial, with this quite high acetic acid in the silage, there was no depression because the inclusion in the diet was relatively low. Okay? So, after 1.7% of acetic acid in the diet, you start to see a depression on intake. Another fact regarding acetic acid is, okay, cow may adapt to acetic acid in the diet. Here we have a control diet and acetic acid diet, a diet supplemented with acetic acid over different weeks. And as you may see, during this first week, the cows received just half of the dose, here the full dose, and after 4 weeks, the cows recover the intake. So they can adapt to acetic acid, but again, it took 3 weeks. Depends on the phase. For instance, for a fresh cow, I think is a big concern. Okay? Then the last part of my presentation is I will share with you a case we faced in Brazil regarding the effect of alcohol in milk quality. How it happened? Ethanol sometimes is used as a substance to temper milk. So it's very strange, but it's still having some people adding water to milk. And then to try to mask the adulteration, one way to do that is to add water. Because the industry detects water in milk, measure the cryoscopy. So there is a normal range of milk cryoscopy. When you put water, you move to this zone. So if you put water plus alcohol, you return the milk to the normal cryoscopy. And then the industry created also a test to detect alcohol in milk. And this is the test. Okay, you put alcohol inside and put heat, and alcohol volatilize and come here to a sulfochromic solution. And then if there are alcohol inside, it turns green and positive. So they do at the dairy industry every day to detect. few years ago, I received a call from a field technician that worked in a big farm, and the situation was the following. The milk tested positive for ethanol, and the thousands of liters were being discarded for more than a week, and they didn't know how to solve the problem. The farmer was losing a lot of money, discarding milk and also paying for the whole batch of contaminated milk because mixed milk with other farmers. And then the dairy industry and the farmer got into dispute because both were stating no fraud. Then how we faced that problem? The first action was, let's confirm that there was no fraud, because both the dairy industry and the farmer was talking there was no fraud. And then we decided to collect milk directly from the teats, and perform the test. And surprisingly, the milk was positive for alcohol. So the conclusion from this first action was there was no fraud because the milk direct from the teats were positive. Both farmer and dairy industry were correct, but the problem continued. Diet may contain alcohols which may impart to milk. And in that farm specifically, corn silage was the sole fermented ingredient in the diet. And then the second action based on that was, let's try to replace the corn silage with other corn silage, for instance, for a small group of cows, using a silage from a neighbor, and retest the milk. And the result was, in the next day, replacing the corn silage just solved the problem. So problem and solution were in corn silage. And then we decided to screen the corn silage and the milk for alcohols. And here you may see different compounds and a relative concentration. So the concentration in the positive silage over the negative silage, the silage that solved the problem, okay? Ethanol is here. Ethanol was a little bit more than twice in the positive silage, but if you look at the concentration of ethanol, even at the positive silage, it's relatively low for corn silage. Okay, so in my view, it was not the case. The second bar is butanol. It's 5 times more in positive than negative, but look, very, very quite low concentration. And the first bar, 10 times more isopropanol. And when you look at the literature, what's the normal for isopropanol? There are very few results for isopropanol, but here some data from our lab. 1-2 ppm, 0.912 ppm, so quite low normal. And when you look there, it's 3,000 ppm. So isopropanol could be the problem. When you look at the milk, the same idea, positive over negative. Ethanol, exactly the same. Isopropanol, 8 times more in the positive milk over the negative milk by the headspace. And then we decided to confirm if the test reacted to the isopropanol, and actually it reacts. Although it's an official method, and the name of the method is, say, ethyl alcohol, it reacts against isopropanol. And even with a higher reactivity, 3 times less, it's already enough to give a positive result. And then we decided to confirm this with a controlled trial. And then we fed 4 different treatments, a control, ethanol, isopropanol, and a combination of isopropanol and ethanol, because we hypothesized that combining these 2 alcohols could potentialize increase the chance for impart alcohol to milk. And then, as you may see, the proportion of milk positive at that test was control 0% ethanol, 0%, because the rumen metabolize ethanol. Isopropanol, 100%, and the combination, of course, 100%. And why it happens? Even at a very low concentration, it's imparted to milk. Okay, look, when the cow consumes isopropanol, it reaches the rumen and it's absorbed. When it reaches the liver, in the liver, isopropanol is oxidized to acetone. Acetone returns to the blood. Part of acetone returns to rumen. In the rumen, acetone is reduced again to isopropanol. Isopropanol is absorbed. So a single carbon chain can cycle different times. So this is why a quite small concentration in the diet can cycle within the cow different times, and part of this can be imparted to milk. So you can see here in our trial, both acetone and isopropanol were increased in the treatments where isopropanol was supplied. So my final remarks. Ethanol, 1-propanol, and 1,2-propanediol, based on the available literature, did not impair intake and performance. Isopropanol can have negative impact on the dairy chain, depending on the country, if this type of test is used. And in my view, we should start to look at the acetic acid concentration in the diets. Okay, the concern is how to measure this at the farm level. Thank you for your attention.
Thank you, João, very much for this in-depth presentation about the silage alcohols. It was very much new information for me as well. So it was very informative, but I'm sure there is still some more topics to discuss about the alcohol issues in silage. So please use the opportunity.
Thanks, Joe. Dave Davies from the UK. Your data with your feed intake with time from your PhD thesis, I think?
Sorry?
The data where you showed your intakes with time and the effect of acetic acid, I think it was your PhD from 2011. Do you think, did you measure acetic acid with time during that? Is it the loss of acetic acid that is having some ability for the cows to improve intake with time after feeding?
Yeah, but the loss is not quite significant. So it's a combination of decreasing the concentration and increase the hunger.
Okay. Thank you.
Yes, acetic acid. So you showed also the data that after 3 weeks the cows adapted to high acetic acid level and the feed intake was not different. So from farmer perspective, what does it mean for the farmer? It means if the farmer all the time feeds the silages high in acetic acid, it's not a problem because all the time the cows get this diet. Animals all the time get this diet. The problem could be only if the farmer decides maybe to change or to use different silage inoculants. For example, homofermentative silage inoculants, and the silage will be low in acetic acid. And some silages are treated with heterofermentative inoculants, and they are high in acetic acid. So, and in this case, maybe if I change one silage to another silage, then it could be the problem. But if I have all silages high in acetic acid all the time, year by year, maybe it's not a problem.
Yeah, thank you, Ivan, for the question. The first point, I think, is I think there is no need for really high acetic acid silages. Okay, we may have good aerobic stability with a moderate concentration of acetic acid. We do not require too high, because if you look for the break point is in the diet 1.7. For instance, if you have 50% of silage in the diet, it means you may have a silage with more than 3% of acetic acid. It's much more than enough for the aerobic stability. So we don't need much more than that. Okay? And the second, perhaps more related to the question, is acetic acid concentration is almost impossible to predict in the silage. It's almost impossible. Every time we will change, every silo.
Hi Joe, Gordon Marley from Alltech. Thank you ever so much for a very informative presentation. The data that you put up from the study, the meta-analysis that you did showing 1.7% acetic acid in the diet, reduced intake, that was screened data for well-fermented silages. Unfortunately, a lot of what we come across is not always as well fermented, and the acetic acid is not always from heterolactic fermentation. So you end up with other undesirable byproducts in the silage that can negatively impact intake. Did you do a meta-analysis on unscreened data to see at what point generically you start to see a reduction in intake?
Yeah, in that meta, there are 10 studies, 3 studies where acetic acid was added, pure acetic acid, and 7 studies where heterolactic bacteria was inoculated. Most of them are corn silage, so no pure fermentation. I'm not sure if I understood exactly the second comment, but you were talking regarding byproducts.
Were you able to look at any data where the hygiene would come into question, where the acetic acid wasn't just produced by Buchneri, whereby it was more of a perhaps real farm environment on some of the smaller farms that are using inoculants?
Yeah. Again, I'm not sure, but in this case, the acetic acid was derived from the heterolytic bacteria because most silage were very typical in terms of heterolytic fermentation.
Okay, thanks. Maybe we move on, and it's Horst next. And
yeah,
still in the back.
Thank you. Horst Auerbach from Germany. I would just like to add something to the discussion about the use of whatever type of inoculant on the fermentation pattern. And João clearly said, and I fully support you, it is extremely difficult to control the concentration of acetic acid by whatever kind of inoculant, except there's one thing every farmer can do, and that is applying the inoculants at the right range of dry matter. And I mean, if you look around in that room here, including myself, there are quite a number of silage additive producers, and I think we have a very big responsibility in advising farmers to use the right type of additive. And whenever we have dry matter levels of less than 30%, a farmer should better keep off his hand from heterofermentative lactic acid bacteria because of excessive or potentially excessive acetic acid formation in the silage. On the other hand, solving the problem by using homofermentative lactic acid bacteria might be a way, but then this comes at the cost of aerobic stability. So I think just focusing on the fermentation pattern is just not enough. We need to focus on quality of silage, aerobic stability from production, cutting basically until the dairy cow or the beef cattle have finally taken that silage in as a part of total mixed ration.
Yeah, just I fully agree. And based on my experience, every time you use a heterolytic bacteria with less than 30%, you have a huge acetic acid and other fermentation.
Okay, I think we have time for one last question, so please go ahead.
All the feeding data you've shown that's based on the silages which were fed and the analysis of the silages. But has anyone ever taken a step back and looked at the actual forages which were used to make those silages? Because in those forages, surely the ratio of fructose, glucose, pentose sugars would vary. And this could have an effect on the production of acetic acid as well.
Yeah, I think so. But anyway, for each comparison, you have the same forage used for the control and for the treated.
Yeah, okay.
Okay?
Okay, thanks. Thanks for the lively discussion. Yeah, and still, we continue.
Acetic acid is the best silage, yes?
The what?
Acetic acid is the best silage.
Is the best?
Is okay? No. Oh no.
If acetic acid is the best?
A high level of acetic acid in silage is not good for intake. It's not good for losses.
In my view, we need a certain amount of acetic acid in the silage because we need to protect the silage against aerobic stability. But we need moderate amount in the silage. We do not need excessive amount of acetic acid. Okay?
By chemical analysis, it's only acetic acid. We don't know divided which fermentation it is. For example, we prepare silage treated by acetic acid, 6 liters. Yes, and the silage was very stable. And the diameter was higher. That at the second silage, which was without additives and bad technology, long cutting and low pressing and higher diameter. And when we tested how much acetic acid, both silage have the same content of acetic acid, but different diameter. And that is big problem for the farmer, how to determine the quality. Because last time we would like the higher content acetic acid by Lactobacillus buchneri, yes, but we determined the same acetic acid. But we don't know the other product, secondary product in the fermentation, and we determination of only acetic acid, yes, but acetic acid mean losses, yes, and last time stability, yes, and that is problem how to determine the different quality with the same content of acetic acid.
Yeah, first, acetic acid, only look for acetic acid is not enough to judge the silage quality. So we need more information to judge the silage quality. So on the other hand, as you mentioned, acetic acid production, of course, there is a price for that during the fermentation, but you also have a protection of the dry matter after silo opening. So there is a compensation for that, depends on your management. This is why, in my view, it's better to have a moderate amount of acetic acid to avoid high fermentative loss, but also to protect the silage after feed out.
I think that the silage which is treated with acetic acid, yes, the fermentation is decrease, yes, and total acid is lower, yes, but when you don't use any additives, and do you have bad technology, you tested only acetic acid, lactic acid, but I think that when is the bad fermentation, there is also secondary fermentation, and secondary other stuffs which can decrease the intake.
Yeah. In this case, you basically restricted the fermentation. I think there is the study from Pekka Huhtanen clearly showing the effect on restricting or the extension of the fermentation on silage intake. It's very clear. It's not based on only one parameter, but the whole fermentation.
Okay. Thanks for the lively discussion. Now I think it's time for us to move to the glass house to look at the posters, and at 1 o'clock there will be lunch in a restaurant just nearby, so I think we will go there together then at 1. But now let's enjoy the posters and continue the discussions. Thank you everybody for the interesting session for this morning. Good morning, everybody. Welcome to the second day of the International Symposium on Forest Conservation. And we will start with the program. Our first speaker will be Davies. He's the director of Silage Solutions Ltd in UK, a very well-known silage person. So let's be ready to listen to his talk and then hopefully have again a lively discussion afterwards. So the floor is yours, Dave.
Thank you, Marketa. Is it working? Yeah, okay. Good morning, everybody. I'd like to thank the organizing committee, especially Václav, for inviting me here to speak today. And I'd also like to thank the hotel for waking you up nice and early so that you're ready for my presentation. I'm going to talk about the silage fermentation and how the fermentation products are utilized or fermented in the rumen. There's a lot of biochemistry, but I hope to put some practical aspects in as well. Because I think often in the silage world, we actually overlook that second fermentation in the rumen. And I'd also like to thank Joe for setting the groundwork yesterday with some of the things I'm going to talk about today. So fermentation and dry matter losses really are important. They have significant effects on the nutritive value. Talk about the silage and rumen fermentations and their impacts. And this is a quote that I've started to use quite a lot. It's from an old text from 1954, a book called The Silage Fermentation. And the author in the preface said, "Any narrow treatment of the matter, meaning silage, from the point of view of one particular branch of a pure or applied science is impossible. And a dissertation on the preparation, characteristics and use of silage is bound to be somewhat discretionary in nature." And what I think he meant was that if we focus too heavily on one aspect, other things change, and we have to take it as a holistic approach when we're looking at silage. So, the consequences of fermentation dry matter losses in the silage clamp, well, there are two aspects to this. They're what I call the direct effects, so the loss of CO2 and water due to an inefficient fermentation, representing an energy loss. But the one that I think really does get overlooked are the indirect effects. So not only when the fermentation produces water, but because there's a significant amount of water in our forage at the start of the preservation process, when we have dry matter losses, we actually concentrate up the water fraction. We concentrate up the undigestible nutrient fraction, resulting in further energy losses and potentially intake losses. So what do I mean? Well, this is a study I did a while ago. I haven't done the statistics because I had a problem with the untreated treatment within this study, and without that I think it's not good. But I wanted to just highlight this to you to get across some of the concepts I'm trying to get through. So percent dry matter losses, and when we're looking at direct losses, this fermentation, the mixed homo-hetero fermentation, had 2.75% dry matter losses versus a homofermentative inoculant or chemical additive at 1.5% dry matter losses. Now, within this, I actually did the silage experiment in a slightly different way, in that I stored some of every sample that went into my mini silo for a pre-forage analysis. So it was a direct comparison of that pre-ensiling forage with the post-ensiling forage. So we can actually look at the effect on dry matter. So with that mixed homo-hetero fermentation, our dry matter dropped from just above 33% to just below 31%, whereas with those that had less fermentation losses, we had a lower decrease in the percentage dry matter in those silos. And that's important when we're considering our lower dry matter preserved forages. The other thing I said was that we increased the level of undigestible nutrients. So to look at this, I've looked at the NDF and the ADF, and we can see, again, with our mixed homo-hetero inoculant, we've had an increase in NDF and ADF relative to the other two treatments. And again, this is driven by those dry matter losses, because it's the digestible organic matter we're losing. And so within this, if I'd measured lignin, I would have anticipated an increase in the lignin content. And I'm going to come back to that later. Now, when we're looking at the silage fermentation, we've got all these microorganisms involved, whether they're aerobic or anaerobic, or on the interface, the facultative anaerobes. And really, the only beneficial one, with the possible exception of the propionic acid bacteria, are the lactic acid bacteria. So nothing's changed over the last 50 years. We still need to have a rapid fermentation to switch off the degradation of the nutrients, either that driven by the plant's own enzymes or those driven by the undesirable microflora. And the best way we can do that is get the pH down as quickly as possible. Otherwise, we end up with the secondary fermentation, Enterobacteria, saccharolytic Clostridia, and then the proteolytic Clostridia, reducing the quality of that silage. And I want us to bear this in mind for the rest of the presentation. So let's just look at what's happening during the silage fermentation. And I'm going to use the commonly used analyte, gross energy, which, as was very eloquently pointed out yesterday, is not necessarily useful when we look at the fermentation in the rumen. But if we look at that in the first instance, and I've associated the reference with every slide, I'm going to go through all the biochemical reactions here from the lactic acid bacteria, and this is from what we normally call the homofermentative, but strictly speaking a facultative heterofermentative lactic acid bacteria. But this is the homofermentation of glucose fructose to 2-lactic, 0 dry matter loss, gross energy loss of 0.7. Please bear with me, there's a few of these to go through. So, obligatorily heterofermentative. One option is a lactic plus an ethanol plus carbon dioxide. 25% dry matter losses, gross energy loss 1.7. One that we more commonly associated with the heterofermentatives is the lactic plus acetic plus carbon dioxide and water. Lower dry matter loss, but a greater gross energy loss. One that we think about less often are 3 fructose plus a water going to lactic, acetic, plus 2 mannitol plus carbon dioxide. On an energy basis, this looks very good on a gross energy basis at 1% loss, and not too bad on a dry matter loss, but that's face value, and we will come back to that later. An alternative but similar pathway, glucose plus fructose, same end products, same dry matter and gross energy losses. Now, I said about targeting a rapid pH decline. The end products from those fermentations, when we're producing ethanol, propan 1,2-diol, mannitol, they're not acidic. So they're not contributing to that pH decline. They're not contributing to the acidification and the improvement or the reduction in loss of nutrients. And then when we consider the lactic and acetic acid, lactic is 7.76 times stronger in its ability to acidify our silage than acetic. So again, even if we produce acetic, it is an acid, but it's not acidifying the crop as quickly as the lactic. So this is a hypothetical fermentation profile. So what I've done here, and I accept that none of these fermentation pathways are solely happening in the silage. It's a mix of all of the fermentation pathways going on. But if we assume that we take sugar very efficiently, drop the pH to pH 4 with our homofermentative fermentation process, and then we compare to all these other fermentation processes allowing for the same amount of sugar to be utilized as was in this, these are the pH declines that those pure fermentation pathways would cause. It's calculated value. So we can see the best one is the lactic plus acetic plus CO2, dropping the pH to about 5.4. Some of them, where we've got the mannitol fermentation, very poor in that pH decline. So, if we're not dropping the pH down, we're allowing a greater growth, a more extensive growth of things like the enterobacteria. And if we look at the enterobacterial fermentations, look at the top one there, 2 glucose, 2 lactic, acetic, ethanol, CO2 and water. That is just as efficient as our heterofermentative fermentation. Yet we wouldn't want to promote that enterobacterial fermentation in our silos. The other two are not as efficient, but if we consider the butane, 2,3-butane-diol has very good antifungal properties and will improve aerobic stability. If we consider mannitol, mannitol is actually a sugar alcohol that is preferentially used by certain enterobacteria. So if we produce mannitol in our primary fermentation, we could actually be selecting for enterobacteria. And many years ago, Ostling and Lindgren inoculated selected species of enterobacteria into model silos and found that they got an improvement in aerobic stability. So if we're promoting these less efficient fermentations through the use of inoculants, is our benefits in terms of aerobic stability actually coming from an increased fermentation from the enterobacteria rather than the heterofermentative lactics that we put on that crop? That's a question that I think we should be answering. Now I'm going to move on to the secondary fermentation products of the heterofermentative species. So 2 lactic, produce 1 acetic, 1 propane, 1,2-diol plus CO2. Again, a large dry matter loss, a relatively small gross energy loss. And then there's a tertiary fermentation where the propane 1,2-diol can be fermented to propionic acid and propan-1-ol, a further dry matter loss, a very small energy loss. Now, the dry matter losses are important because they're the dry matter losses that are contributing to those indirect losses in our energy in terms of increasing the relative proportions of the undegradable fraction. So this is a mini silo experiment, just focusing on the gross energy from the lactic, acetic, and propane 1,2-diol. Done at Harper Adams University in the UK. Kate is in the audience. If you want to talk more about it later, I'm sure she will help fill you in. Maize silage trial, 2 homofermentative additives, the same additive, 50% rate, 100% rate, and then these 2 are not commercially available, and then 3 of the leading heterohomofermentative lactic acid bacterial inoculants on the market. Significant improvement in lactic with the homofermentative, as you'd expect. Significant improvement in acetic acid with the homohetero, but actually our untreated silage here has as much acetic as those 3 products. And then looking at the propane diol, as you might expect, an improvement with some of the hetero-homo mixes, but not the others, not the other one. Now, using the gross energy values just from that silage fermentation and calculating the gross energy values of the lactic, acetic, and propionic jointly gives us this result. So, a significant improvement or a significant reduction in the amount of gross energy lost through that homofermentation. Now, when we look at that, that was 0.2 megajoules per kilogram dry matter. So if your cows are eating 14 kilograms of silage, that equivalent is a loss of 0.5 litres of milk. So that's part of the story and part of my concerns when I hear we can improve the energy content of our silages through the production of some of these secondary and tertiary end products, particularly propane 1,2-diol. But we can't look at the silage fermentation without looking at the rumen fermentation. So when we look at the rumen fermentation of lactic acid, it actually requires 2 hydrogen ions to produce that propionic acid. And actually, by absorbing that hydrogen into that fermentation pathway, we're reducing methanogenesis because hydrogen is the main source of methane. So actually, we're capturing energy that would have been lost. And so when we look at the gross energy change, there's actually a positive, and we gain 12.7% theoretically. And by removing hydrogen ions, we're actually increasing the rumen pH, or we're absorbing hydrogen ions that react is a measure of pH decline. So this is some work from Pekka Huutonen when he was at SLU, and quite nicely shows that feeding sucrose versus feeding lactic acid to the rumen, lactic acid has a higher rumen pH. There's actually less lactic acid because it's efficiently fermented to propionic, significant improvement in propionic. And then when we look at microbial protein synthesis, no difference between sugar and lactic acid, implying that the rumen bugs can grow just as efficiently on that lactic as they can on the sugar. So again, we hear quite frequently that high lactic silages cause a rumen acidosis. The published information would suggest otherwise. Now, I've talked about the lactic and the change that happens with lactic in the rumen. When we produce acetic acid, that's one of the major end products of the rumen fermentation. So there's no gross energy change within that fermentation pathway when it hits the rumen. But, again, like we heard yesterday, some of that acetic acid, particularly in hot climates, will volatilize. So if we've lost it before it gets to the animal's mouth, that's 100% loss of energy of the acetic acid that's been lost. When we look at ethanol, in our primary fermentation, that was more energy efficient than the acetic acid heterofermentative fermentation. But when that hits the rumen, it's very rapidly fermented to acetic acid, which has a gross energy loss of 36%. So we're actually losing energy, even though in the silage fermentation on our gross energy values, it implied that we weren't. Propane 1,2-diol, again, if that's fermented in the rumen, goes to propionic acid, but produces hydrogen ions. And that production of those hydrogen ions results in a gross energy loss of 12.7%. So again, it's not so simplistic as just looking at the silage and the gross energy losses within the silo. We have to consider the whole picture. And when it comes to the case of mannitol, I think we need to do a lot more work. Because Ahmed et al. In 2013 found that mannitol was very slowly fermented in the rumen. It's very water soluble, so it's likely to flow out very quickly from the rumen in the liquid phase. It's also hydrogen rich, so is it going to increase methanogenesis from the rumen if it is fermented in the rumen? That's a question. But in the human health industry, mannitol is used as an artificial sweetener because it's not absorbed very well from the gut. What happens in the ruminant? If it's not absorbed from the gut and it escapes fermentation in the rumen, then all that mannitol that was actually being calculated as very good in terms of losses of gross energy, very low losses of gross energy, could actually become very big losses of gross energy. We cannot take the silo or the rumen in isolation. So we need to put these fermentations together. So when we look at hexose sugar going through the homofermentive pathway, 2 lactics, we produce 4 propionic, 4 water, gross energy gain because of that reduction in methanogenesis of 9%. Hexose through either of the acetic or ethanol fermentation pathways in the silo actually end up with the same end products in the rumen, gross energy loss of 8.8%. Hexose plus 2 hydrogen ions going through the secondary fermentation pathway where lactic went to propionic plus acetic, propane 1,2-diol plus acetic, assuming that the primary fermentation was homolactic, gave an energy loss, gross energy loss of 26%. So like I said, we focus too much on certain aspects. We need to consider the whole aspect in terms of what the animal can utilize. So, experimental evidence. I think we need much more. I think we need to focus some of our attention on some of these factors that I've brought up from a theoretical perspective this morning. But this is some work published by Navarra-Villa. I haven't got the statistics on it because I've pulled out 3 treatments from 8 treatments. It was actually done in Porrick Akaile's lab, and those of you that know Porrick, he liked to do everything in one experiment, but I wanted to simplify it. So, in terms of acids, this was the total acid, homofermentative, lactic, acetic, so we can see that the homo-hetero mix had as we might expect, a much more acetic fermentation, untreated intermediate, and this one much more lactic. Rumen pH, silage pH, 3.6 up to 4.1 in the silo. In Rusitec, went from 6.67 to 6.7. I've put it on a large scale, but there's no difference between those. When we look at rumen acetic acid concentrations, much higher with our acetic acid concentration from our homo-hetero, very similar with our untreated homo-fermentative only. Rumen propionic, showing that lactic goes very rapidly to the propionic acid in the rumen, much higher levels of propionic acid, much lower levels of propionic in the homo-hetero mix. Sorry, Simon. This is the dry matter digestibility. So we can see here I've included the pre-ensiled crop, untreated homo-fermentative inoculant, significantly higher than our hetero-homo inoculant. And that was actually a 3% loss in DOMD due to that indirect losses or indirect concentration of the undegradable fraction. Rumen methanogenesis. When we convert this on a basis of total rumen VFA, because total rumen VFA is our utilizable energy, we can see that our homo-fermentative inoculant had lower levels of methanogenesis than our more acetic fermentation there in the hetero-homo fermentation. Now that's not going to be just down to the acetic acid, it's going to be down to the fact that there's more undegradable fiber in there. On farm, I come across additives that have been used on farm in inappropriate conditions. This is a farm that had a problem with intake. Maize silage, I analysed, the analysis I show was done on the 12th of February. It was ensiled the very beginning of October. I decided I was going to do a wet NIR, the UK system, dry NIR through one of the satellite labs of America, and then proper chemical analysis. I was actually surprised how close the lactic and acetic were to the wet chemistry. But what I always find is that the chemical analysis compared to the NIR analysis, NIR over-predicts lactic and under-predicts acetic. And that are the analysis that farmers get. They get one of these NIRs, which is generally much less accurate than I would like on those key VFAs. Now, you can see the other fermentation products. So ethanol, quite a significant fermentation to ethanol. Propanol, again, quite a significant fermentation. The dry NIR lab was predicting 7.2 grams per kilogram propane 1,2-diol, whereas the wet chemistry didn't find any. So we need to be careful when we use lab-based NIR analysis when we're looking at the fermentation end products. Now, you'll probably say this was too wet. It was treated with the mixed heterohomo that's claiming lots of energy from 1,2-propane-diol. It actually had 34% starch. So when we do a back calculation from the losses, based on the fermentation profiles, and this was 3 sample dates over a time period from November through to February, 9% fermentation losses I calculated in that silage lamp. If we value that, and sorry, I've done it in British pounds, for every 100 tons, that represented £1,224 of lost income, just on those dry matter losses. And I've also back calculated the dry matter of the original forage, and it was actually 33%, give or take, because obviously you can't specifically say which fermentation pathways they've gone down, which fits very well with what I would expect from the starch content of that silage. So I think when we're looking at the holistic approach, we need to look at everything and not just one aspect when we're thinking about our silage additives. Now, we heard a lot of debate about intake yesterday. Well, there's some quite strong data showing an effect of NDF, higher NDF, reducing intake. When we have losses in our silo, fermentation losses, we're increasing the NDF content. I'm not going to explain this because it was far better presented yesterday by the original author. But again, debate about the effect of acetic acid. But we shouldn't focus on one aspect. It's the NDF, it's the acetic acid, it's the dry matter. They're all going in the wrong potential direction to improve our intake. There are many animal studies with homofermentative lactic acid bacteria showing a positive effect on animal performance. Areola et al. 2021 conducted a meta-analysis where they looked at the obligately heterofermentative lab in silage inoculants, giving no performance benefit over no treatment. But in that paper, they did highlight a tendency for a negative impact on maize silage. But they did admit there were not enough studies. So again, a hole in the data set. We need more data. Whereas Oliveira, earlier, again, a meta-analysis, same group published these two, showing a benefit in terms of silage fermentation and homofermentative lactic acid bacterial inoculant. So in conclusion, there are many interactions occurring in the silo, the silage fermentation that can have an impact on nutritive value and animal performance. And whilst homofermentative in the silo has been highlighted 30 years ago as causing an issue for aerobic stability, it has a very positive effect on the things that drive animal production. So, additives do affect what happens, and I think we have gone too far with our focus on aerobic spoilage because we can see it in the silage lamp on farm, and we don't see all these additive effects that are having negative effects on intake, negative effects on animal performance, negative effects on the carbon footprint of ruminant agriculture. So I think we should work harder to find other solutions rather than thinking we already have it. Thank you for your attention. Questions?
Thank you very much, Dave. That was very fundamental and very much needed. Let's discuss. What do you think? Please raise your hand for the microphone.
Thank you very much, Dave, because according to me, it's a very important part of losses. Calculation, losses dry matter, and losses of the gross energy, because farm cannot calculate it. Yes, and you calculated losses of money, and they cannot calculate it. I think that this point is the most important for the not only for fermentation in silage, but for fermentation in the rumen. Yes, and thank you very much for it.
Thank you, Václav.
Dave, thank you for this very fundamental and specific biochemical excursion. My question is, what is your opinion to anti-nutritive things like esters in maize silage or biogenic amines? What is the role of those in dry matter intake?
I think they have a very important role. I think they should be a greater focus of attention. I know there are one or two people in the room that have a serious focus on this. Biogenic amines are formed as a result of protein breakdown, and I think there's not enough evidence, but I would suggest that there is evidence showing that more rapid fermentation will improve our protein preservation, reduce the effect of biogenic amines. And in the cheese industry, biogenic amines are part of the flavor development. And the longer the preservation of that cheese, the more biogenic amines are produced. And biogenic amines are produced by heterofermentive lactic acid bacteria in order for them to be able to survive low pH. It's one of their survival mechanisms. So I think there's a lot more work that needs to be done on that. In terms of the esters, again, and I'm waiting for the person sitting next to me to possibly contradict what I'm going to say. There is some complex biochemistry there that I think we need to understand in greater detail before I could properly answer that. But you wouldn't be surprised if I think homofermentatives are going to be better than heteros.
Hey, I was thinking about one step here in between the silage fermentation and rumen fermentation, and the silage fermentation produces different amounts of fermentation end products, and they have different volatility.
Yeah.
So there is one step there in between that you might lose some before they end up in the rumen.
Yeah.
So how significant is that, and how would that change some of the conclusions here?
Personally, it's something that I really want to look at, but living in Wales, it's not the best environment to look at volatilization, because today's a really nice summer's day in Wales. But if we look at the gross energy losses, acetic is going to be one of the main ones, ammonia is another one. They're going to be lost, so it is a greater loss. So in terms of the effect on animal intake, it might be lower, but in terms of the effect on the loss of energy, it could be higher. Again, Joe touched on it yesterday, so maybe he should comment on the volatility more than me, because he's probably got a more experience.
Do you want to comment?
Thanks, Joe.
Yes, as I mentioned yesterday, we carried different trials even during the summer in Brazil, and most of the volatile were actually ate by the animal because although they are volatile, the rate's not so fast, even in a warm climate.
Yeah.
So the numbers are, the loss is 10 to 30%. The rest is consumed in a warm climate. Here, perhaps, the loss is even lower.
Okay, thanks. Okay, what do others think? We still have a few minutes time to discuss.
Last question. You spoke about Clostridia, yes? And I would ask you if you tested or analyzed the biogenic amines, because it's not normal analyze, yes, but in Czech Republic sometimes with alfalfa and the grass, it's a lot of biogenic amine, yes, a secondary fermentation, and that is problem because the analysis is very expensive and normal we don't make it.
I have, on occasion, measured biogenic amines. I haven't done enough work, and I have to say, Václav, I think the Czech Republic is probably the best place in Europe to be measuring biogenic amines, but maybe Germany as well. So there are limited capacities, and the lab I go to is a commercial lab that sometimes I wonder whether the samples are being treated correctly, but it's something I think I'm very interested in. I think there needs to be a lot more work because these secondary end products from either a good or a poor silage fermentation are probably having much more effect on the animal than we actually credit them for. And it's something we should be looking at. But I also think we need an on-farm reliable method for the measurement of acetic acid rapidly.
Okay, hey, one more topic. I interpreted your presentation in a way that the heterolactic fermentation is not maybe the best solution to control the aerobic stability. So there would be these chemical additives that could be added, and then the effects on the fermentation would be smaller. So what do you think about that?
Whilst I didn't totally agree with Horst's comment yesterday, because I don't like heterofermentative lactic acid bacteria, I think the selling of the inoculants has to be much better done on farm. So you must not sell the mixed species product on low dry matter silages. I personally think that the combination of chemical inoculant or chemical only would be a much better approach. But again, I think we need to look at the carbon footprint. I would be quite happy, as many in the audience know, if heterofermentatives were banned from being used on farm, but I'm in a minority.
It's not a question, it's also a comment. I want to say thank you for showing the comparison between the analysis of NIRS and chemical analysis between, I think we should have the focus on the limitations of the NIRS, and we have to discuss it again and again, and to look very intensive which data were presented. That's why thank you for this.
I totally agree. I think NIRS is causing quite a lot of problems on farm for fermentation quality.
Okay, thank you, Dave, once more.
Thank you.
Oh, sorry, was there still? Okay. Okay, so then we continue with the second presentation of this session now before the coffee break. And I would like to invite engineer Jiří Janoušek from Brno University of Technology to present us some different type of aspect regarding silage production. So go ahead, Jiří, the floor is yours.
Ladies and gentlemen, good morning. Thank you for the opportunity to present our research regarding to quality monitoring of corn crops using multispectral camera on the UAV, on the unmanned aircraft. My name is Jiří Janoušek. I am from Brno University of Technology and especially from Faculty of Electrotechnical Engineering and Communication. Before we get to actual topic, I would like to start more broadly, because since my topic links to agriculture with modern technology, it's necessary to reflect what is precision agriculture is. After that, I would like to follow with sensors and technology, what we used, and describe the sensors. And if will be time, I would like to finish my presentation with other aerial sensing applications and how we apply this precision agriculture and can help you with this. What is precision agriculture? Precision agriculture involves treating different parts in the same fields in the unique ways. That allows farmers to divide their along the more granular lines than ever before, and we use square meters in strategic. Traditional methods of precision agriculture and nutrient spraying are wasteful and not cost-effective. With the ability to capture more precise measurement of individual fields, including from ground-mounted sensors, has allowed us new era of smart farming and to take a hold. When farmers can perform precision agriculture, spraying based on accurate data, crop yields and production, and go up while waste these lines. What are benefits of precision agriculture? Without economic, with economic aspects, is very important for us because efficient of use material inputs, and we are able to not function at all. It is reduction of material inputs or maintaining the parameters that you are put into efforts, not but only increase the yields. Obviously, without functional economy, it's not going to work because it's more important parameter of our future in the environment. Environmental, but we have to comply with the standards and parameters. Therefore, intensity of the applications, you should be friendly of our environment. Last but not least, is added value to makes work easier and saves your time. In the picture, you can see differences, two states difference. It's clear that is in the past fields were approached more individually on this picture. But today, when we feel farmed, heavy machinery is used for high efficiency and it's necessary to enlarge these fields. But it's necessary to find some compromise for that. Efficiency, how to manage with the least possible workload, and how to least approach this situation more than 70 years ago. The aim of precision farming or precision agriculture is not to take on parcel or land of a unit, as you can see on the left side, but to take in account is homogeneity, definitely. Our aim is to look on the whole area and where each part is can be given special care, something like on the right side. Every field is not the same, definitely, especially small differences on the start cultivation show us big differences on the results. I think in the picture you can see nitrogen rates and edit. On the left is homogeneous edit, and the right actual condition are taken in account. We strive to achieve work process that allow us to continuously improve our results. It starts with the first phase, where we collect data from UAVs, from some sensors, and we evaluate our collection data, examine what the results are. And the last phase is applying the results and testing them in actual practice. It's very important for us. We take our results every year and compare with the new results, definitely. Because this allows us to increase accuracy and decision making over again. To achieve this, the modern technologies that you may be familiar with other applications, but haven't that you're using in agriculture, I think. For our approach, our basic element that we used is some platform. Platform, it means that we use drones that carry where they attach some sensors or cameras. Drones can either be from something like these wings. We have with disadvantages should be covered a largest area that because longer flying time. Or other types of multi-rotor copters, they're later servers of local area, I think. But can also cover large areas, about more than 100 hectares. It's important to mention that nowadays is really anyone can control this. It's not required to any deeply long study and how to use it. Because it's really to control it. The imaging and cover the whole process, the whole territory. Drones are not controlled manually. The flight path is completely planned before in the application. You can simply draw some polygon, some area that you want to map, and that set altitude, that set your camera, and application will create a whole path and whole flight plan before, according to the camera angle and scanning your speed. Everything is automatic, definitely. It's not longer because you can click and you can start directly flight. This graph shows us spectrum of light, because humans can only see that this small part of visible spectrum or whole spectrum. This is visible radiation that we see. That is why, as someone tells me, some farmers tells me that I see everything. I see on my field everything, but I will say that definitely not see everything, because there is a little bit larger area that you cannot see. There is another important light spectrum bands that we can see with special sensors, special cameras. Especially for us, it's really interesting thermal infrared or infrared radiation, which is great influence for health of plants. In our research, we focus on short waves of this area of our spectrum, from 800 to 1 micrometers. That is also a reason that thermal imaging camera sensors are very expensive. We use a multispectral camera which does not see the entire bandwidth, but contains 5 separate, really narrow separate spectral bands. And it starts with visible spectrum with blue, green and red band. And therefore there is a red edge. It's about 700 nanometers. And this is important component that near infrared band, because the near infrared band is the one non-really longer in the band where the most differences plants are. The camera works on principle that measuring reflectance sunlight and falling surface between these wavelengths. In the differences, reflectivity between individual zones are the building blocks that of vegetation research. The reflectance of, for example, water or some soil, whole entire bandwidth is almost zero, but contrast reflectance of plants is increased in near infrared band. Is much more in the visible bands of reflection. Proportional vegetation indices are formed in the monitoring zones. Indices take account with percentage changes in individual zones. For example, normalized vegetation indices is made up to ratio of NIR, it's mean near spectral band, and red component. The fact the health plants, or for example green plants or green leaves, have higher reflectance than plants that don't have such a high chlorophyll content. Ars not handily is used for the plants reflectance value decrease of near spectrum bands and indices for the red component. It seems simple, but unfortunately health of the plant is amount of biomass or is affected by many other factors such as lack of nitrogenous substances, pesticides and another chlorophyll and amount of precipitation. It's very difficult to determine which spectral components are suitable for and need specific in cultivate crops. This is the reason that why more 100 vegetation indices are currently published. Some are very similar because they used some similar spectral bands, but comparison of differences in spectral bands is our camera can capture. It's set to analyze and compare 3 vegetation indices. Start with NDVI, green NDVI, and NDRE. On this picture you can see each of individual bands on the left side that are bands that our camera can capture as monochromatic images, but is already clear from what vegetation differences are in reflectance. For example, soil, you can see soil and other plants. Entire maps of the reflectance are combined for whole maps, for whole images, and it's precisely selected vegetation index what can be observed together. On the left side you can see a field created by RGB camera and with visible spectrum, and on another images you can see maps show larger differences what it shows. It's all of them clearly show some paths from heavy machines, but there are bigger differences from another indices. is the reason why we compare these indices. Imaging does not have only provided with camera on the UAV, on the unmanned aircraft, on the drones. It can be combined with satellite images, I think. It's allowed to capture multispectral data too. One of the frequently used are satellites. In the picture you can see comparison of multispectral camera and mounted on drone, and on the right side is the satellite image from the sky. At first glance you see higher resolution of the left compared with the satellite on the right. I think is the main advantages of this, of why we use drones. But every situation has some pros and cons, I think. The advantage of unmanned aircraft are the ability to zoom some specific area what we interested. But satellites flies over every 2 days, 3 days, I think. And it's, if its weather is good, because if it's cloudy, the specific spectral bands is not will allow pass through the clouds or pass through the rain, I think. The advantage of satellites is cover larger areas than drones, but it's idea to combine both, definitely, because satellites initially determine basic variabilities, and using with drones we set to focus to local, accurate, real-time measuring. There is a satellite image, no matter how advanced camera is in question may be, but still from the orbit. Drones flying over field and delivers specific and great proximity image resolution, I think. Furthermore, when farmer use satellite images, the picture may be in real time, maybe a day old. Drone can provide more up-to-date informations and allowing even greater precision regarding to the pesticide or what you need. But I come to the topic itself, which is monitoring the quality of agriculture crops using real-time multispectral drone imaging. As I mentioned, the whole process is seems simple, I think. At Gregor, we during the whole intervals growth of corn, we now taking images with drone with multispectral camera in each reflectant spectral bands. We create from this our vegetation maps for more indices. If you selected 3 indices for comparison. But the main problem that we are solving, that the changes over time are under spectral reflectance. We get only relative values from this. This is why during drone imaging is manually sampling of corn is also performed simultaneously. Samples that we are able to ground into corn are subjected to chemical analysis simultaneously, and the results of the chemical analysis, the vegetation indices are correlated together. The results are the ranked multispectral indices map and with exact chemical compositions values of corn silage. We establish linear, equations, some linear prediction models that further refineness of each area. For shooting, we use mentioned multispectral camera with automatically capturing and calibrating panel, and panel is used for accurate expose calibration in different sunlines, wavelengths. Here is example of the parameter of the size image field. in area about 13 hectares, I think. We images from height of 40 meters, but most commonly is to taking images from 100 meters altitude, and we are able to possible more larger areas. Our experiments were conducted in several locations of South Moravia region because we want to start a bigger variation of conditions at the same time, and several maize hybrids were usually tested. On the right pictures you can see photos of each multispectral bands where you can see Václav Kár how to sampling these corns. Here is sample of values from 1 year for one corn hybrid. There is 8 samples of corn analysis what were determined, and for example there is dry matter content and another parameters that are interesting for us. Some curd protein, starch, ash, fiber digestibility. The most important parameter for us, I think, is clearly dry matter content, definitely, because it's a parameter for determining harvest time. It's usually a harvest time window for this hybrid is between 380 to 420, and this corresponds in this case with 5 round of stage with the sample. This map shows us one spectral band from multispectral camera accurating images on which we have 2 samples of corn hybrid as this marked, and the highlighted window you can see the sampling of location. This graph shows us individual reflectance of each band that we are captured. All the maize hybrid studies show us these linear proportions and larger differences in the reflectance for the spectral components of NAR spectrum for visible us. For these bands it should be the same reflectance, but in this case we can see bigger differences with NAR spectrum. The vegetation indices are calculated with obtained reflectances, and for 2 hybrids we calculated 3 observed vegetation indices: NDVI, NDRI, and green NDVI. This table shows us comparison of these values, because the chemical analysis data and vegetation indices values was correlated together. We have 5 corn hybrids, and the correlation is done for all hybrids separately. We have 5 hybrids together, and for them we have nutrition analysis with strong parameters that show us this correlation. To make easy understand it, we have 3 vegetation indices for each hybrid, and this correspond correlation value. We can see how correlation is strong. This table, this correlation together of whole 4 years show us that results that is strongest correlation for dry matter content across the vegetation indices. Thus our hypothesis is confirmed that dry matter content is more important parameter for us. To verify that our hypothesis works, we specify some linear regression models for dry matter content. You can see values of dry matter content on the x-axis, and allow vegetation indices to be calculated and given dry matter value. That means that the farmer it can be able to immediately determine vegetation index value obtains with dry matter content, and not waiting for longer process that otherwise leads to this. We have been doing this research for 4 years, and during this time we have already had opportunity to test some how to verify our results are function. Here is field that contains I think 12 hybrids of corn with the narrow bands, and we have one mark hybrid. We carry out our experiments the day of the harvest and just before cutter went it. The snapshot was taken with the sampling that we made evaluation for and correlation. This is a table. This is table when you can see 5 hybrids of corn that we tested, and the actual value of analysis, nutrition analysis, in the first row. This first row is sampling nutrition analysis. And with our regression models that we created, we compare our NDVI or NDRI and green NDVI indices, and that we found that vegetation indices for green NDVI on the smallest variation. There is smallest variation. The question may come up, what are results, what is imply of this our harvest time prediction? Green NDVI vegetation in this is has strongest correlation with dry matter content, but is necessary to calibrate multispectral camera before each flight to obtain the best results as we want to be. Differences between hybrids should be trivial, but it's only necessary to determine the equations for stay green hybrids. But to precisely capture of harvest time window that we want to reach the exact harvest time, it's necessary to take images with drone or with satellite every 4 days, because the window is really narrow, I think. Is everything from our research, but I have some another sensing applications if we have time. You can see differences between another applications where multispectral camera can be used. This is we start with RGB photo. There is no big differences what we can see. NDVI index show us big differences before each field. It's not homogeneous, definitely, because we are able to mark critical points. These images more similar with satellite data, and we are able to create some application map from our NDVI or NDRI imaging. Definitely there are some benefits of multispectral imaging, because we are able to get plant health analysis. There is some potential where we calculated together with Václav of how to are able to save some chemistry, potential increase yields, and definitely we have some significant environment impact. We focus not only multispectral imaging, but we are able to create some another applications with UAVs, with unmanned aircrafts. there is some examples what should be do it. Classification of some areas that we are specific, some problems we are able to evaluation what each point on the map it's mean. We are able to create 3D models, precision, I think, precise 3D models, because this example of one farm is precision 3D model where we calculated bigger differences with the silage pit. We are able to calculate amount of each silage pit. We are able to calculate it and recalculate it directly to the dry matter content and precisely estimate the value of these blocks. There is some other applications. I think it will be using maybe in future, not yet using some spraying with drones, because these drones now can carry on only about 20 liters of some liquid. I think a small piece for larger areas. But interesting for us is that we are able to create some elevation models. Elevation models means that we are able to know precision altitude of these fields, of these areas. Another application should be interesting for some counting of animals. With thermal imaging, we are able to calculate how many animals are on the field, or finding some secret places that you are not see animals directly from on the ground. There is, I think, last example of using thermal imaging. This is vineyards on South Moravia, near to Mikulov city, and there is example, really deep precision example with thermal camera that you are able to use. This is a hill, and this vineyards is situated to south, and you can see how these trees helping cooling these vineyards on the left side, and how the hill is this hill has bigger temperatures, about 60 degrees. There is a little bit more example of Václav how to calculate temperature of the silage directly with the thermal camera on the ground. Thank you for your attention. If you have any questions, you can ask.
Thank you very much. It was really interesting to see these applications. Is there some questions or comments from the audience?
You showed a table with the 8 stages of sampling. So can you explain the reason why digestible of organic matter decreased so much from stage 4 to 5? And the second question, I saw in your data of the correlations in 2020 negative correlation of digestibility, and in 2021 there were positive correlations. Can you explain that?
Yeah. It's caused because these hybrids is not the same, definitely, and it's caused that this hybrid is stay green hybrid, and there are another place that from another hybrids, because it's one parameter that I not mentioned is weather, definitely. Weather causation for us, differences in rain, we have big differences with soil, and this caused that these correlations are cause big differences, I think. It's mean that we want to evaluate these regression models for each hybrid, but not important to set for each hybrid directly, but we are able to create two groups for green stay hybrids and non-green stay hybrids. This correlation shows us that this is function, but there is a lot of problems with weather, and we should evaluate how the weather influence this correlation. We not yet do it. And
did you answer both questions? Yeah, okay.
No, it's okay.
Yeah, okay. Hey, I was thinking about the other applications that you could do. Do you think it would be possible to use drones to take samples from the fields, if it's fields far away or difficult or save labor?
Yeah, definitely. Your University of Prague already do it. They taking samples from ground directly, because drone can reach really deep precision position of these samples. They create some image recognition for some plants from another, and they are able to take a sample directly from ground with unmanned aircraft.
Okay, that could be quite useful for farmers and experiments as well. Some more questions or comments?
Please translate. Má vliv výška porostu a hustota porostu na výsledky a případně jak počítáte tu korelaci od...
The question is
pan přednášející mluví česky, takže...
The question is how height of plants influence the correlation indices, how influence the vegetation. Yes, definitely has influence, but this stage of corn, it's mean for us that each sample, when we evaluate this correlation, cause that values increase or decrease in the first stage, and the last stages should some parameters decrease, I think. Definitely height of plants influence the multispectral imaging. Is main task how we calculate these equations.
Okay, and then?
At first I would like to say thank you for this very nice and attractive presentations. I think it's very important for the future that such a young generation has to work on it, and I am very happy that you are working on it, and I encourage you to work on it. I have a question. You have many nutrient content on the table. Do you know a project to investigate the mycotoxin content on the field, DON, zearalenone, T2, or aflatoxin? Because in the case of maize silage, the aflatoxin is very important problem nowadays, and when the summer is a little bit wet, in that case the DON and ZEA and T2 also can be a problem. It would be very useful to detect the level of contamination on the field.
Okay, Václav, you can explain this, why you evaluate these parameters, and
I would like to show you the first picture.
regarding to the question, I think.
No. First.
Okay.
I would like to show you the picture.
First picture?
First picture, university. I hope that somebody is remember for this place. This place is 4 years ago. Our silage meeting was in this place, yes, and we start together, cooperated with Mr. Janoušek, yes, and the results are only for this year, for 4 years only, and we plan another activities, yes, but we open a lot of questions for it, yes, for example, how many plants, for example, diseases, and so on. But we choose these based parameters, yes, and we test it if we take some correlation, yes, and I think we will continue to work, yes.
Thank you. Some more things to discuss? I guess not, so thank you once more. And now it's time for the coffee break. Okay, time for coffee break. Thank you. And then we will be back here at 11 for the next presentations.